just so that I don't forget. You should see a sign pop up on top of your screen. Do you see that? Saying that the session is being recorded?
Yes.
Yeah, okay, thank you, thank you. So just do a thumbs up and that's fine. If you don't want to turn on your audio or video, you can do that too. So just let me know that you can hear me. Thank you, Madeline. Thank you. And so you guys can see the form. Those of you coming in, joining a bit later may not be able to see it. So I may request some of you guys to just forward it. I'm going to post it again. Okay. But throughout the session, as people are joining in, I will ask those of you who joined earlier to share the link. Okay. So you can just put it back on chat and people can click on it and save it on the devices. Okay. All right. Okay, let's give 1 more minute and then we will get started. I am recording the session. So those of you who are here and can click on the link and get the form, please do a thumbs up so I can see. Okay, thank you, Tyler. Thank you, Jennifer, Xiomara, Sinjai.
Yeah.
Okay, thank you, thank you. Okay, it is going to be a long session and very intense, so I plan on, can I share the review packet? Who's your professor, Kim? Let me see if it's gonna let me share the file. Let me try. Okay. Let me see if it's gonna let me share the file. Okay, looks like it's not letting me connect to OneDrive. Hold on, see if there's any other way I can share that.
Thank you.
Okay, I'm gonna need some time to look it up. I shared it with my
Professor.
class.
This,
A different class.
this is, yeah, this is just for,
Okay, I will need to download it from my Blackboard to share it. So in the meantime, if somebody else can do that, I will appreciate it. Hold on.
Yes.
Okay, let me try sharing this one. Okay, try and see, Hygiene. Try and see if this is, you can open this, okay? Can you open it? Kim, can you open it?
Yes, thank you.
You're welcome. Okay. All right, then. Let's get started. So what I am going to do, and I posted a link for the form, attendance form. So please make sure that you fill it in, okay? Okay, did everybody get that?
Oh, yeah, sure. Thank you.
Okay, you're
So
welcome.
what is the attendance form? Excuse me.
This is, the link is in the chat and you should have gotten it from your professor. If not, somebody in attendance now can put it back in the chat so that those of you who are joining now can open it, and just click on it and it will be on your device, okay?
I don't see anything in the chat for attendance. I'm sorry.
Okay, so maybe you joined in after I had posted it. So
Maybe, yeah.
yeah, so if anybody else can do that, hold on, let me, before I actually get started.
Okay, I see now.
Do you
I
see
see.
it?
Yes.
Okay.
Thank you
All right.
very much.
Thank you, Mia. Thank you very much. Okay. All right. So let's get started.
My
And
job is to make college easier because students have a
actually, Mia, I don't know what you posted. It looks like a YouTube link. I don't know what that
Yes,
is.
that is a YouTube. Sorry.
Okay. So.
Oh, I'm
would you-
sorry. I copied and pasted what you had. I'll try again.
Okay. No, that's not what I had. I think it was on your device. Hold on. I'm going to try again. Okay. See if this is going to work for you. Okay. It says forms.office.
Yes, now that is
Yeah?
the right one. Yeah. Thank you
Okay.
so much.
Okay. All right then. Okay. So let's get started. And before we get started, some of the ground rules. You have you attended? Is this the first time you're attending a review session, or have you attended it before? The first one for lab exam 1.
Yes, before.
You did. Okay.
Mhm.
So just know that the packet that we are going to be using, and I have actually shared the file. You should also have it. I will be sharing on screen the review packet. That is just a guide. So if you look on top, it says Rockville Campus Lab Exam Review Guide. Okay,
Mhm.
review guide for lab exam. So don't try to race with me to write down word for word. It is being recorded, so you can go back and listen to it again. But my main goal is to make sure that you understand the concept. It's not going to be copied and pasted on the exam. Okay, the format is exactly the same as your first lab exam. It will be the same format, that part of it will be going to stations, answering questions from the station. You will have pictures or data or information that is there, and questions in your exam that you are going to take and answer those questions, come back and complete rest of the questions on the exam. Okay, so that will be the format, and it's up to your professor how your professor is going to do it, whether they want you to go to the stations first and then work on the exam, or whatever way they decide. But that's going to be the same, and the exam is going to be the same for everybody. Okay, everybody taking BIOL 150 exam 2 at Rockville campus, it will be the same exam. Okay, some of the questions may be slightly different, but the station question will all be the same. So what you should focus on is to try and understand the concept, so that when you read the exam questions, you can understand what it means, and you can do the calculations, you can answer those questions. Okay, does that make sense?
Yes.
All right then. Okay, so what I am going to do is I'm going to share my screen, and I am going to start with the packet. Okay, can you guys see it or not?
Yes.
You see the screen. Okay, so you can see the review guide.
Yes.
Yes.
Yeah. Okay, all right. So what I am going to do is I will give you some concept, basic concepts before we answer the questions. And I like to use whiteboard quite a bit, okay? And we are going to start with the enzyme lab. So we will go in the order of the review packet, okay? So exercise 7 is the enzyme lab, which is very intense. There's pretty long of your almost 20-page document, 50%, almost 50% questions from the enzyme lab, okay? Questions from the enzyme lab. And then the rest half, or maybe 75%, is going to be on the PCR lab that you just finished, okay? So these two labs are both very intense depending on how much time it is. We will take a break maybe towards the end of the PCR lab, and then do transformation lab and mitosis. We'll finish with mitosis, okay? So we will go in the order of how it is organized, not according to the schedule, okay?
Okay.
All right then. So these concepts, should we have covered all of these? These concepts should be easy for you, what enzymes are and what are they made of, okay? Can someone tell me what enzymes are and what are they made of?
Enzymes are proteins. If I'm not
okay.
wrong, correct me if I'm wrong, they're proteins that are made up of amino acids.
Exactly. So enzymes are made of amino acids, they're proteins, but what are they?
Aren't they proteins or?
They are, the molecule is protein, most of them. There are other types of enzymes also, but they are made of proteins. But they have when we ask what are enzymes, we are talking about the function.
Catalyst.
Catalyst.
Catalyst, yes. So they are biological catalysts. Okay. Does that make sense?
Can you repeat that again, please?
Yes.
So enzymes are made of proteins, and their function is to speed up the rate of a reaction. Okay, they are made of amino acids. Does that make sense?
Yes.
Okay, so what I am going to do is can you guys see a whiteboard?
Yes.
Okay, so here is a concept map that I am going to be using. Okay, so enzymes are biological catalysts that speed up the rate of a reaction. Okay, they speed up the rate of a reaction. Okay, so enzymes are very, very specific. They can break down specific substrates. So enzymes speed up the rate of a chemical reaction, the kinds of reaction that take place in cells. okay? So they are made of proteins, okay? They can lower what? Because when a reaction is taking place, it needs some energy to overcome what?
Activation energy.
Mhm.
Yes.
Activation.
So if a reaction is taking place, okay, if this is the energy for the reactants, and this is the energy for the product, when the reactant or the substrate, they get converted. So the reactant gets converted into product, okay?
Mhm.
Enzymes or catalysts can speed up the rate of the reaction. This conversion of reactant into product can be sped up using a catalyst, okay? There
Mhm.
are other ways to speed it up also is by heating.
Mhm.
But in living cells, this is not an option, okay? What living cells do is make these kinds of special class of proteins that can act as a biological catalyst. Biological catalyst, okay? So these reactants, when they get converted into product, they don't get converted into product just like that, okay? They require some energy so that they can get to an unstable state called transition state before they get converted into product, okay? So this amount of energy that is needed, that is called the activation energy, okay? Now, do enzymes provide that energy? This is the energy, this is free energy. Energy, do enzymes provide that energy?
Yes.
Yes.
The answer is no. Enzymes do not provide the energy.
Oh.
Okay. What enzymes do, this is the amount of energy when enzymes are involved. Okay. Enzymes lower the activation energy requirement. Activation energy requirement. Okay, they do not provide the energy. So this much energy is needed without the enzyme. This much energy is needed. So this much energy is needed if enzymes are added. So much less energy is needed. Okay, if enzymes are added. So enzymes do not provide that energy. What they do is they lower the requirement so that there's less, they can get to the transition state much easier if enzymes are added. Okay, so that's what enzymes do. Okay, what I am going to do is let me look at some of these questions.
I have a question.
Yeah.
I'm sorry. Can you just
No.
reiterate where you said they lower the requirement of energy and what happens after that?
They lower the energy that is required. Okay, they lower the activation energy requirement.
Mhm.
So it's not, it's freezing up. Let's see if it's gonna let me open it. Okay. Hold on. Sorry. Okay, so what I'm going to do is I'm going to share some of these links with you and how enzymes work. Okay, so this is the one that I want to show you. Should be playing here. It's not. Okay, so this is a really nice one that kind of will summarize how enzymes work. Okay, and I will share with you where you can find that information also. Okay.
I think we can hear anything.
You don't hear anything?
No.
No.
No, we can only see.
You can only see. Okay, let's see if I can switch to, hold on. Okay, let's try now. See if you can hear. Can you hear now? Can you hear now?
No, still not.
Still not, okay. I think maybe because I am. Okay, let's try this. How about now? Were you able to hear now?
No. No, still nothing.
Still no.
Mm-mm.
Okay. Huh. That is very weird. Okay.
The audio.
Mm. Okay, one last time. Otherwise, I'm going to give you the link and you can watch it and we will go through the explanation. Okay.
I think somebody mentioned that you need to mute yourself in order for us to listen the video. I don't know.
You might need to stop sharing your screen and then start sharing it again and then hit the option to share audio.
Okay. You know what? I am going to not play it. Can you hear me?
Yes,
Yeah, we can hear you,
I
but
will share the links. Can you guys hear me?
yes, we hear you.
Can you guys hear me?
Yes.
Yes, we
You
hear
can
you.
hear me. Okay. So I think maybe, I don't know, something is not working with the YouTube speakers. I am going to get back to the whiteboard and explain from there. Okay. So the activation energy barrier that I was talking about, this is the figure that I'm talking about. Okay. So can you see the energy and the reaction coordinate when the reactants get converted into product? Okay. This is called the energy barrier. This graph that you see here, this is called the energy barrier. And if there are no enzymes, it needs a lot more energy, activation energy without the enzyme. It needs a lot more energy than if we add enzyme, it needs much less energy. Okay. Enzymes speed up the rate of a reaction by lowering how much energy is needed. It does not provide the energy.
Makes sense.
Okay, does that make sense?
Yes.
Okay, so that's what I was talking about, that how do enzymes speed up the rate of a reaction? Okay, so if you look at your packet, enzymes increase the rate of a reaction by do you see anything? It should be a drop-down menu.
We can see it. I can read
You
the
can see it.
options.
Okay,
Yeah.
okay. So for some reason, something's happening with my computer. Oh, okay.
There.
Yeah, okay. So it's showing very differently than how it does on my office computer. Okay. Well, now I know what to do. Okay. So yeah, so enzymes increase the rate of a reaction by
Lowering.
lowering the activation energy requirement. Okay. So that would be, does not provide activation energy, It does not provide any ATP. It does not generate heat. Okay, so none of those are true. Okay.
Mhm.
Or can you leave it a little bit? I wanted to take note quick.
It should be on your, you have the packet, right?
Mhm.
Okay, so in your packet, it is fillable document. So if you click on it, see this little caret that you see? If you click on it, you will see all of that option. Okay,
I don't s-
and you can select from there. Do you not have the fillable document?
No.
You don't. Who's your
No.
professor?
Professor Cosgrove.
Professor Cosgrove. Okay. I am gonna.
I have the packet, but I don't see the different option you're talking about.
So you don't see this fillable document?
No, I only see an answer and a line. That's it. And then
Okay.
we have to fill.
Okay, that means that you don't have the fillable version. Okay, so I just posted it in the chat, and you should be able to get it from there.
Oh, okay.
Okay, so you can open it up on your device. That way you will have this. You can type it in and you can fill it in. Okay.
Okay.
So what are enzymes and what are they made of? Enzymes are made of proteins. They are biological catalysts. What is the importance of enzymes in living systems? Why are they important?
Chemical reactions.
Because there are thousands of chemical reactions that are taking place in the living cell. Within each of the cells, there are many, many reactions that are taking place. Okay. And if there were no enzymes, all of these reactions would take forever. And some of these reactions may not even take place. So having enzymes speed up the rate of those reactions, allow living organisms to live, to do everything that we are doing. Okay. Do you think high fever for a long period of time would alter cellular function?
Professor, can you please repeat your answer for number 2?
Number 2, what is the importance of enzymes? Enzymes speed up the rate of all reactions, chemical reactions that are taking place in the living cell. Okay.
Okay, thank you.
The reactions would not take place without the enzymes because it will take a very long time for those reactions, or some of the reactions may not even take place without the enzymes. Okay. And organisms would not be able to do the things that they are able to do, like live, process energy, grow and develop, cell division, all of these things, everything that living organisms are capable of doing, it's possible because of the chemical reactions that are taking place inside the cell. Okay. And so every reaction is catalyzed by one or more enzymes inside the cell. So living cells would not be able to live without enzymes catalyzing the reactions. Okay. Does that make sense?
Yes, it does, yeah.
Okay. Okay. Do you think high fever for long periods of time would alter cellular function?
That's a very good question. I think it can do. I think it can affect, or maybe I'm wrong.
It can.
There's a possibility because enzymes are affected by temperature, and especially that high temperatures that make the nature the enzymes.
Yeah.
It can yes, that is correct. If there is high temperature, because what happens with high temperature? Remember that they are made of proteins. If
Yes.
proteins are heated, what would happen to the proteins?
They will be destroyed.
They denature.
Yes, it would be denatured. Okay, it would be denatured. Okay. Every enzyme has a specific substrate. What determines the specificity of an enzyme? Specific substrate meaning the reactant. In biochemical reactions, the reactions that take place inside the cell, we call them biochemical reactions. Okay. So the reactant is called substrate. Instead of calling it a reactant, we call it a substrate. Okay. And they get converted into product. every enzyme is specific for what substrate it can convert to product. It's not going to take any substrate and convert it to any product. They're very, very specific. Okay, so that's what it means, that every enzyme has its specific substrate. What determines the specificity of an enzyme?
Structure of the enzyme.
Structure of the enzyme and structure of the substrate as well. It's kind of like a lock and key kind of mechanism. You know, you can take a lock and you can take a key. Just because you have a lock and a key doesn't mean that the key is going to work for that lock, right? It has to be that particular key that is going to unlock that one, right?
Yeah.
So
Okay,
enzymes and substrates are like that. They are specific for each other.
Oh, yeah, that's true.
Okay, it's not
So-
going to work with everything.
Is it, can you say it has a specific active site for a specific substrate?
All
Specific
enzymes, okay, so that's a good point that you brought up. Active site is something where the substrate binds, okay? The shape of the active site is such that the substrate, the active site is on the enzyme. The shape of the substrate is complementary to the active site so that it can fit in nicely, okay?
Okay.
So for example, let's say that this is the enzyme and this is the active site of the enzyme. Okay, this is the active site of the enzyme. And there are this kind of molecules, this kind of molecules, and maybe this kind of molecule. Okay.
Mm-hmm.
This one is kind of close to that. Okay. So this one
Mm-hmm.
may be able to fit here. Okay. So let's do it in a different color. Let's see that way. Okay. And let's say that this will fit here. Okay. At the active site. Okay.
Mm-hmm.
And this, when it fits, it can get converted into product. Okay. So this place where it's sitting, that is the active site. And this is the enzyme.
Mm-hmm.
Okay. So this would be the substrate that gets converted into this product. This is the product. So once the enzyme works on it, and it will kind of close in on it, induce fit, and convert it into product, and this becomes that product.
Mm-hmm.
Okay.
So, excuse me, Professor. So you say that. So what determines the specific of enzyme is the structure of the enzyme and also the
structure, the shape of the active site and the shape of the substrate also. And it will bind covalently. Let's do a different color. Covalently bind at multiple points with the amino acids at the active site. Okay, so the substrate will temporarily bind to the active site while it is being converted into product. Okay, all of this you probably have covered in chapter 8, 9, okay, in lecture. We talked extensively about what enzymes are, what are they made of, how do they work. So it's the same concept, but we are studying a specific enzyme as an example. Okay, so in the enzyme lab, what enzyme were we studying? What is the name of the enzyme?
amylase.
Amylase enzyme. Okay, so we were studying the amylase enzyme. So I'm going to use that as an example. What was the substrate for the enzyme amylase?
Also,
What was the substrate?
the starch.
Starch, yes. So the substrate was starch. Okay. And you had water, it was in a solution of starch, right? And what did enzyme amylase do to starch? What is starch?
Polysaccharide.
It's a polysaccharide, yes. And what kind of a polysaccharide?
Carbohydrates.
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 Okay, so glucose is what?
A sugar.
Sugar.
Is it a sugar? Is it a carbohydrate?
Two.
No, it's
Yes.
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 Yes, it is.
Is it a polymer?
Yes.
Yes.
Glucose is a polymer?
No.
No, monomer.
Monomer, okay.
Oh.
And starch is a?
Polymer of
Is it?
glucose.
Yes. So starch is a polymer of glucose, okay? Starch is a polymer of glucose. Glucose is a carbohydrate. It's a simple carbohydrate. Starch is also a carbohydrate. There are many different kinds of carbohydrate, okay? So starch is a polymer of glucose, meaning that many units of glucose are linked to make this, okay? Make this polymer, okay? So many of these glucose units are bonded together. And what amylase is going to do is it's going to break the bonds. What is the name of the bond? The glycosidic bond is hydrolyzed by the enzyme amylase. And what do you get? What is the product?
Maltose.
Maltose, yes. And maltose is what? Is it a carbohydrate? Is it a protein? What is it?
Saccharide.
What is it?
Disaccharide.
Disaccharide, yes. It's a disaccharide made up of glucose and glucose. Okay, 2 glucose units together. Okay, so this is the substrate. Starch is the substrate. And maltose is the product. Okay, so if this is the enzyme amylase, the product would be maltose, and the substrate would be starch.
Maltose. Sorry, starch. My bad.
Okay. So what is amylase doing? Amylase is converting starch into maltose. Okay. Okay, going back to okay, I'm just for some reason it's locked up on my thing. If you keep an enzyme at 0 degree for extended period of time and then bring back to its optimum temperature, would you expect the enzyme to be active?
Yes.
Okay.
Or no?
No, that is correct.
Yes,
Okay.
it's correct.
Yes is correct. So if take a look at this diagram here, effect of different conditions. Okay. So this would be the y-axis is the dependent variable enzyme activity, and x-axis is the independent variable temperature. So this is showing the effect of temperature. Okay, this particular graph is showing the effect of temperature on enzyme activity. Okay, why would temperature have an effect on enzyme? Typically, with increasing temperature, activity would increase. Okay, activity would increase with increasing temperature. Okay, but notice how here it starts to go down. This is the activity scale. It starts to increase and then it starts to go down. This temperature at which it has the highest activity, what is that called?
Optimal temperature.
Temperature.
Right. Temperature at which enzyme has the highest activity, that is called the optimal temperature. Okay, so all enzymes have these optimal conditions. This is the effect of pH. Okay, so this is the pH at which this has highest activity, this has highest activity, this has highest activity. So these enzymes, different enzymes, have different pH at which they have highest activity. Okay, so for this enzyme, this is the optimal pH. For the yellow one, this is the optimal pH. For the green one, this is the optimal pH. Okay, so all enzymes have their own set of optimal condition, optimal temperature, optimal pH, optimal salt concentration. They have their own set of conditions at which they have the highest activity. Okay, what is the activity of enzyme? To catalyze a reaction, conversion of the substrate into product, whatever that enzyme is. Okay. So if you keep an enzyme at 0 degree, okay, at 0 degree, you are keeping an enzyme. When you work with the enzyme in the lab, the enzymes were stored at either in the fridge or at -20, okay, at -20, which is freezing. But then when you used it for your experiment, did the enzyme work?
Yes.
Yes. Okay. So when we keep enzyme at a lower temperature, the enzyme remains structurally the same. The structure does not change. When it is losing its activity here, activity is going down, that's because the overall 3D structure of the enzyme changes with increasing temperature. Because it's a protein, it becomes to unfold and denature. Okay. So all the bonds and interactions that keep it folded in the specific 3D shape start to fall apart. And the shape in which the shape of the active site and the overall shape of the enzyme becomes distorted, and somebody needs to turn off the
Hold a handstand for 10 seconds, get $50.
Can you guys please mute your microphone? Yes, thank you. Thank you. So this enzyme, no. Someone
I'm wondering if it's the video to play.
Yo, Genesis, mute your mic.
Okay. All right. So the enzyme, the overall 3D shape of the enzyme is going to change in such a way that this substrate is no longer either going to fit, or it's not going to fit in a way that the enzyme can make the change. It's not going to convert it into product. Okay. Depending on how distorted the shape of the active site is. This enzyme is going to change its shape when the temperature or the salt concentration or the pH is changed. Okay. In your exercise, if you look at your exercise here, talks about the effect, the activity of the enzyme is affected by different conditions. Okay, different conditions, factors like temperature, pH, salt. And so you need to know what are the factors that affect enzyme, the structure of the enzyme. Anything that affects the structure of the enzyme is also going to affect its activity, which is its function. Okay. So the shape of the enzyme, the 3D shape or the structure of the enzyme, if that is affected, it is also going to affect the activity. Okay.
So going back to the question, if it's a zero, will be active, right?
At 0,
enzyme will be active.
The enzyme will not be active because not enough temperature to cross the activation energy barrier. It's still lower temperature. Okay. So it still needs some temperature to become active, but the shape will be the same. Okay, shape is not going to be affected.
Oh, okay.
As you increase the temperature, now it's getting more and more energy, even if it is very little energy here that it needs to cross the activation energy barrier. Some energy is still needed, right? So it still needs to have this much energy to cross the barrier, okay? This much energy is needed. It is less than what it would need without the energy, but it would still need that much energy, okay? So when you keep it at low temperature, it does not have that much energy. So as you increase the temperature, now it's getting that energy from the surrounding environment, and the enzyme is going to become more and more active.
So for question 7.5, it says if you keep an enzyme at 0 degrees Celsius for an extended period of time, then bring it back to its optimum temperature, would you expect the enzyme to be active? And I know you said yes.
Yes.
And explain because the structure, I know it was denatured and it just goes back to its shape.
At 0 degree, it does not get denatured. Okay, so here, let's do this here. Let's do a kind of a table here. So the two things that affect, okay, the structure and function, okay? Now, when the enzyme is kept at 0 degree, structure is normal, okay? No change in structure. Function is low activity at 0, okay? The reason it is low activity is because it's not enough to cross the activation energy barrier because the energy is low, okay? When you bring it to, let's say, human amylase, okay? When you bring it to 37 degree, the structure is normal, okay? Enzyme activity is going to increase because now it's getting to its optimal temperature, okay? If you get up to 80 degree temperature, the enzyme is denatured and function will decrease because the enzyme is denatured. So here, activity is low, here the activity is low, but the reasons are different, okay? So when you are looking at a graph like this, effect of temperature, okay, and this is the activity, here at low temperature, activity is low, right? This is also low activity, this is also low activity, okay? The reason it is low active is because there is not enough energy.
Okay, I think I get it now. Thank you.
Okay, to cross activation energy barrier, okay? Here, it's low because enzyme is denatured, okay? Both of these areas are where the enzyme activity is low, okay?
Wait, I want to take note of that. What did you write here where you said to cross barrier?
Oh, not enough energy to cross activation energy barrier. Okay, so everything that I will show you also, if you want to kind of cross-check and go back and read up on it, I will show you where you can also find the same information. Here, okay? The activation energy barrier, that it gets its energy, because what happens is when you start heating it, you increase molecular movement. Reactions depend on collision, molecular collision, okay? When you increase the temperature, they start moving faster. They collide into each other, and that allows reactions to take place, okay? When enzymes are there, enzymes lower it by providing a space where the substrate can bind at the active site and get converted, which is why enzymes can speed up the rate of the reaction, okay? So the introduction in this exercise is what I'm talking about right now, okay? Which answers question 7.5. Does that make sense?
Yes.
Okay. Guys, can you please mute your microphone?
Whoever is, yeah, please.
Okay, thank you. Okay, list 3 factors that can denature a protein or enzyme. So the 3 factors:
Temperature, protein, and concentration of salt.
not protein, temperature, pH,
pH,
and salt
is
Temperature,
concentration.
pH, concentration. Yeah.
Right. Okay. So temper factors that affect enzyme activity are also the factors that affect enzyme activity. Okay. So also denature. The reason it affects activity is because it affects the structure. Okay.
So the conditions are is the temperature, the pH, and what again?
Salt concentration.
The salt concentration.
Right. So the reason salt concentration affects the activity is because of the ionic bonds. Okay. The ionic bonds, anything. So here, denature at extreme pH and high salt concentration, because the ionic bonds are affected. Ionic bonds, the tertiary structure of the enzyme or the protein is held by hydrogen bonds, disulfide bridges, ionic bonds, van der Waals interaction. All of these are important for the enzyme to hold its 3D shape. Okay, so anything that opens it up or anything that affects it is going to affect the activity of it. Okay.
Okay.
So the 3D structure is held by different kinds of bonds. Okay. Hydrogen bonds, ionic bonds, hydrophobic interaction, van der Waals interaction, all of these. Okay. So question 7.7, what is the optimum pH of an enzyme? Optimum pH of the enzyme is the pH at which enzyme has the highest activity. Okay, so we looked at this graph, right? So if we change this to pH, okay? So if we change this to pH, at low pH and at high pH, it is, let me use a different color that way. So if we change it to pH, you will see the same kind of graph, except that at low pH, it is also going to be denatured. Okay. So these are now pH. At low pH, it's going to be denatured. At high pH, it is going to be denatured. Okay. And whatever optimal pH is of the enzyme, it's going to have the normal 3D structure. Okay.
So the temperature of the pH, so the optimum temperature depends on what? Sorry, can you repeat the for question 7?
So optimal pH is the pH at which the enzyme has the highest activity. So if this happens to be the pH, this is the pH scale, this pH enzyme has the highest activity, right? So this would be the optimal pH.
So
Okay.
the optimum pH of an enzyme is what again? Repeat that.
Optimal.
The optimal
pH.
temperature.
Optimal pH. It's the same thing. Optimal pH is the pH at which enzyme has the highest activity. Okay, that's what optimal pH is.
So in other words, it varies.
Yes, yes, absolutely. Like if we keep on changing the pH, right? pH is increasing from low pH to high pH.
Mhm.
At low pH, it has low activity. At high pH, it has low activity. Okay. In case of, okay, again, did you guys read the exercise at all?
Yes.
So changes in the pH and salt concentration affects the stability of secondary and tertiary structure that is maintained by hydrogen bonds and disrupt the salt bridges. Okay. And as a result, enzymes denature at extreme pH and extreme high salt concentration because the bonds break up. Okay. The difference is that temperature and pH have a very different effect let's see if I have it here. Okay. When it is temperature, temperature has a different kind of effect, okay? Temperature affects the enzyme activity in two ways, in two different ways. As temperature rises, molecular motion increases and the rate of random collision increases, okay? Forming more and more products as you keep on increasing temperature, but it does not keep on increasing at all times, okay? So when we looked at this graph, you saw the increase because temperature is increasing and molecular motion is increasing and more product is forming, okay? Nothing had happened to the enzyme. But here, activity is decreasing because the enzyme is getting denatured. But when we change it to pH, at low pH, enzyme is denatured, at high pH, enzyme is denatured. At low salt concentration, enzyme does not have its 3D shape. At high salt concentration, enzyme does not have its 3D shape, okay? So whatever the optimal condition is, whether it's pH or salt, optimal condition is defined as the condition at which it has the highest activity, okay?
Yeah.
All right then. Okay, so why is the pH of the environment of an enzyme reaction important? Because it allows it to maintain its optimal 3D shape, okay, active 3D shape. So it needs to have all the amino acids folded, the polypeptide chain folded in the right way, not the amino acid. Amino acid is what makes up the enzyme, but the polypeptide chain has to be folded, the secondary structure, tertiary structure, all of it in the right way so that the substrate can fit in, okay? So anything that affects the 3D structure is going to affect, whether it's pH, salt, or temperature. Do all enzymes have the same optimal pH? We looked at this graph right here, right?
Wait, I missed that last part. You said because it allows it to do what for letter A, question 7?
Which one? Say that again.
The one you just went over. Why is the pH of the environment of it?
Why is the pH of the environment of an enzymatic reaction important? Because pH of the enzymatic reaction of the environment is important because it holds the enzyme in its active 3D shape. It's responsible for maintaining the 3D shape of the enzyme. Okay?
Okay. Are you saying pretty shape?
What?
What did you say? Are you saying pretty shape?
3D.
3D shape.
Oh, I'm so
3D.
sorry. Okay.
Allowed to maintain the 3D
3D shape.
3D shape. Okay.
3D shape or
the-
conformation. These are the words that refer to the overall shape of the molecule. Okay?
Miss Raman, do you have the packet with you? Are you looking at
I do, but yes, I am looking at it, but I have hard time sharing it because it does not let me connect. Zoom is not letting me connect to my OneDrive.
Okay, I was just wondering because I know the last review we had, because it's hard to read on the screen sometimes when you're doing the things.
Yes,
I think
but
it helps if you could type with us like the answer, so we don't have to keep.
This is like a 30-page document. Okay, so I am not going to no 20-page document. I take that back, but it's going to be very hard for me to keep. I'm not a fast typer. Okay, so but I will try to share the document with you. It didn't let me. I did share, I think, earlier. Did someone?
Yeah, I have the document. I was just wondering if it was possible if you could type. We could all
Okay.
answer
So
together.
okay. So the other thing that I have said is don't worry about typing. Know what the information is and where to find it, because we are not going to copy and paste the questions. Okay.
Right.
So for the exam,
understand.
So make sure that you understand what optimum pH means, that the pH at which the enzyme has the highest activity, that's what optimum pH means. And so if you are given a graph, the reason you need to understand that, for example, here you are looking at a graph, right? So this graph is showing the effect of temperature. So if you are asked, what is the optimal temperature of amylase? Okay, so this is showing you human amylase. If you're given a graph, it's going, the kind of questions that we will probably ask is like, based on this graph, what is the optimal pH? Now, if you don't know what that means, you won't be able to answer that, right?
Yeah.
Mhm.
I have a
So,
question.
okay, let me finish with, who
Okay.
was asking me the question? Let me finish with her if she's okay.
Okay.
Oh, I'm fine.
You're fine? Okay, okay.
No, actually, can you restate again? Sorry, because that was a lot of interruptions that I get lost. So you say basically because allows to maintain the 3D shape of the enzyme.
Yes,
Okay,
yes.
got it.
Okay, yes.
Thank you.
So it's a chain of amino acids that fold through the secondary structure, tertiary structure, and then have an overall 3D shape. Okay,
Hey, Professor, do you mind if I say something real quick? I just want to,
Sure.
I just want to go ahead and say this because I said it in the chat, but I want to say this now. This isn't directed at anyone, by the way. But if you guys missed something, you should just go to the recording after and look again because we are only on question 8 and it's been an
Thank you.
hour.
Thank you very
we
much.
should-
Yes.
We should try and,
Thank you.
yeah, we should try and
Yeah.
prevent the
I
professor
forgot that
from
I was
repeating himself.
recording also. Thank you for
Yeah,
reminding
or
Tyler.
another thing is that there's also going to be like other 2 sessions, like on Saturday and on Sunday. Correct me if I'm wrong, but yeah, there's other sessions.
No,
as
that
well.
is absolutely correct. That is absolutely correct. And thank you, Tyler. Thank you very much. Yes, we are recording. And so if you missed something, that is exactly right. You can go back and listen to the audio recording. Your professor is going to make it available to you. Okay. I will share my email address, but it's on the flyer with the announcement, all of the professors, so you can email them directly if you go to their sessions and ask for the recorded session. Your professor is going to make it available. It is not going to be possible for me to send the recorded session to 30 different people, but your professor is going to make it available, okay?
Okay.
Can I ask my question now?
Mhm.
So we did talk about the optimum pH for the enzyme. So would you say that the salt is, I know would be also affecting the enzyme, but what would be the optimum for, I mean, when we talk about salt, like if it's low or
Okay, so
increase,
the
what would you-
graph activity, it does not matter what the condition is, whatever the activity is, you can have temperature affecting it, you can have pH affecting it, you can have salt concentration
Okay.
affecting it, okay? You would
Okay.
get the same shape of graph. Whatever that condition is, the
Okay.
highest activity will be, if this is temperature, then this would be optimal temperature. If this happens to be pH, then that would be the optimal pH that you would read off of the scale. If it is the salt concentration, increasing salt concentration, then that would be the salt concentration at which it has the optimal condition. Okay, optimal
All right.
activity.
Thank you.
All right then. Okay, I think it's time to move on to the next one. Okay, so list and describe. So this is a very important one. List and describe the steps of the scientific method that you use to design your amylase experiment. Now, something that you have done before, right, with the osmosis experiment, right?
Yes.
Yeah. So you should start with what? What is the first step?
Question?
Start with question,
Mhm.
and then
Hypothesis.
Hypothesis.
hypothesis. Yes. What would be the next one?
Prediction.
Prediction. And remember that prediction has to be if and then statement. Okay. Prediction has to be if and then statement. So if you start, for example, we're looking at this. Okay. A question may be, what is the optimal temperature of human amylase? Okay. So we are looking at this. A question could be, what is the optimal temperature of human amylase? What would be a hypothesis? How would you write the hypothesis? You have done this. You have also submitted the report, right? If the question is, what is the optimal temperature of human amylase? That is your question. Obviously, it's going to end with a question mark. Your hypothesis is going to be an educated guess, okay? That will answer this question. So you can say optimal temperature of human amylase is, just pick a number.
37.
Okay, 37 degree. Okay, so that is your hypothesis. Okay, your prediction has to be an if and then statement. Okay, so if the optimal temperature of human, so you are basically copying this whole thing and writing it. If optimal temperature of human amylase is 37, then what? How do we know that that is the optimal temperature? Then
Time activity will be highest.
Higher.
Then it will have the highest activity at 37, right? So that is your prediction. That if the optimal temperature of human amylase is 37, then it will have the highest activity at 37, right?
Yes.
Does that make sense?
Yes.
Okay.
Yes.
Now, how would you design an experiment?
Amylase at different temperatures.
Right. So what you would be doing is doing the same experiment with human amylase in multiple tubes, same substrate, same enzyme, and just do different temperatures. So maybe 0 degree, 20 degree, maybe 37 degree, 45, what you did in the lab, 60 degree, 80 degree, right? So the same experiment at different temperature, right? And then measure the enzyme activity. Let's say that this is the graph that you got. And then based on these results, you either support or reject your hypothesis, okay? Important thing is to identify what is dependent variable and independent variable, okay? So here, when you look at your prediction, you try to be as specific as possible, okay? Typically, the if part of the statement is your independent variable, okay? So this, I'm going to write it with a different color. So the if part of the statement is your independent variable. And the then part of your statement would be your dependent variable, okay? And the reason you want to go that way is because if this is the independent variable, sorry, the optimal temperature, which means the temperature, which means that should be your x-axis, should be your temperature, okay? Highest activity, that is your dependent variable. So that should be your y-axis, okay? So you know that you should have the active, this is what you are measuring against the different temperatures, okay? The way you have your experiment set up at different temperatures, so that would be, the different temperatures would be your independent variable, and dependent variable would be the activity that you are going to measure for each of these, okay?
Got it.
Okay, so you want to write down your hypothesis in a way that makes it easy for you to figure out what is what, because obviously the next steps are going to be question, hypothesis, prediction, experimental design, results and conclusion, right? And then based on what you find, maybe you would find that this one is actually active at 45 degrees. Okay, highest activity at 45 degrees or at 25 degrees and not 37. So based on that, you would say that your hypothesis was not supported by the results, okay? This is why your hypothesis is an educated guess. You guessed 37 because it's human amylase. Our temperature is 37, and that's why you guessed, which makes sense, right? Okay, so here, effect of temperature, 4 degrees, what happens to the structure? So these are the words that you are going to be choosing from: denatures, unfolds, correct 3D shape, properly folded. So it would be properly folded at 4 degrees, okay? Function, it will be inactive or low activity because there's not enough energy at 4 degrees for the enzyme to do anything. But the structure is properly folded. There's nothing wrong with the enzyme, but it's not going to work. It is going to be inactive because there is not enough energy in the environment at 4 degrees. Very little molecular motion, okay?
Inactive.
At 85, the structure is going to be denatured. It will unfold, okay? It will unfold and it will be denatured. Activity, it will be inactive because it is denatured, okay?
Excuse me, the function of 85 degrees is going to be what?
It will be inactive.
Inactive, okay.
Okay. Now, suppose 1 ml of fungal enzyme, question 7.1, 1 ml of fungal amylase enzyme was added to 5 ml of 2% starch. Solution was kept at 45 for 15 minutes. Okay, so you have 1 ml of fungal amylase that was added to 5 ml of 2% starch. So let's draw this out. So you have a tube where it was 5 ml of 2% starch, right? And fungal amylase was added. So this has starch and fungal amylase was added and it was kept at 45 degrees for 15 minutes, okay? So what do you think happened in the tube? The starch
Maltose.
Converted to maltose, okay? Which is what? Which is a disaccharide, right? Now, if you took this after 15 minutes, you took this and did a Benedict's assay. Anybody remember Benedict's assay?
Yes.
Okay. So if you did the Benedict's assay with this, would it be positive or negative?
Positive,
Yes, it would be positive. Why?
because it has a sugar.
No, not because of that. What is the Benedict's test for?
Detect the disaccharide.
Sugars.
Sugars.
Reducing sugars.
Reducing sugar, yes. Benedict's test is for reducing sugar, okay? And maltose is a reducing sugar, okay? So the reason test will be positive is because maltose is a reducing sugar, okay? So this maltose is a reducing sugar. So when you do the Benedict's test, it will be positive because maltose is produced, okay? Do you think the mixture would produce a color change if heated with DNS? We heated with DNS, would it change color? Yes, it would change color. Why? Both of these, Benedict's test and DNS assay, both of these are tests for reducing sugar. They will change color if there is a reducing sugar. Okay, so maltose will change, so this will change color of the Benedict reagent. This will change color of the DNS reagent because maltose is a reducing sugar. Okay.
So we can say the reason why we change its color is because maltose is a reducing sugar, right?
Exactly. That is the explanation. Yes.
Okay.
Okay. Why did we use DNS assay instead of Benedict's assay? So since both of these, and you have done Benedict's before, why did we use this and not this one? The reason we did that is that Benedict's reagent, when it tests positive, it makes an insoluble red precipitate. DNS assay, when we did the DNS assay, it will produce a color change, but it's homogeneous, okay? And we use spectrophotometer to look at the change in color, okay? So DNS assay produces a soluble product. Soluble product. In Benedict's assay, it is insoluble product. So the entire solution does not change color. Okay, entire solution doesn't change color. It stays kind of at the bottom like this, maybe a little bit, but very uneven. But here, the color change is very even. Okay, the entire tube will change color depending on how much maltose is produced. Okay, that's the reason why we use DNS assay and not Benedict's assay. Okay. You decided to perform a BIOL assay on the enzymatic solution. You observed that the solution tested positive. Why? So you took this solution and did a BIOL test.
It's for proteins, right?
Is it
What?
because it is a protein?
What is a protein? Which one?
The billet acid.
The amylase.
Right. The tube has starch, which is carbohydrate. Amylase is a protein. And when you do the BIOL test, that is testing for the protein. Okay. Whether it is functional or not, it's not going to tell you whether the enzyme is active or not. It will just tell you that there is a protein in this tube. That's all it will tell you. Okay. So you decided to do BIOL test on the enzymatic solution. You observed that it tested positive because amylase is a protein. Okay. It's a protein. BIOL test is a test for protein. Okay. Does that make sense?
Yes.
Yes.
Okay.
Yes.
All right. Let's look at question 11, 7.11. The calculations. Make sure that you can do the calculations and know how to do the conversion. 350 microliter is equal to how many ml? If you convert it into mils.
3.5.
3.5? Are you sure?
35.
35, yeah.
Yeah, 30.
0.0350.
0.035.
Oh.
1,000 microliter is equal to 1.0 ml. Okay. 1,000 microliter is equal to 1 ml. So 350 microliter is equal to 0.35 ml. Okay. So if you have 500 microliter, how much is that?
0.5.
0.5, yes, 0.5 ml, okay? 350 microliter is equal to 0.35 ml. So make sure that you can convert back and forth. If you are given microliter, you should be able to convert it into milliliter. If you're given milliliter, you should be able to convert it into microliters, okay? Okay, looking at the table here, I am not going to do the entire table, but make sure that you are able to calculate the concentration. So this is given in milliliter. This is what you have to figure out, okay? Now, when you do these types of calculation, again, this is a formula that will not be given to you. C1 V1 is equal to C2 V2, okay? This is the formula that you should know. And from the problem, you should be able to figure out what is C1, what is V1, what is C2, what is V2, okay? So when you get a problem like this, write down what is C1, what is V1, what is C2, what is V2, identify that. This is what you will need to know. This will not be given to you, okay? So in this problem, C1 is 5 mg per ml, okay? V1 is the second column, maltose solution, whatever solution is given. So let's say for tube number 3, okay? So for tube number 3, what is the volume?
0.4.
Okay. So what you need to find out is C2. Okay. The total volume V2 is 10 ml. So it tells you in the question that the solution per tube after was 10 ml. Okay. So now you need to solve for this. C2 is equal to C1 V1 divided by V2, right? So that would be, make sure that you keep the units, otherwise you are going to make a mistake. Divided by 10 ml, okay? And you just solve for that, okay? What is that equal to? Somebody has a calculator, do it quick and should be something
0.2.
0.2.
0.2, sorry.
0.2 mg per ml, right? So mils and mils will cancel out, okay? So 0.2, yes, okay? So this is a formula that you should know and know how to use it and how to convert it, okay? Yes.
And the 0.2 would be for 0.4 maltose solution?
Right. So
Okay.
you are, when you are using this as the V1, you are doing it for tube number 3. So you will need to figure this out for each of the tubes.
Right,
Okay.
okay, thanks.
Okay. So when you do a standard curve, okay, how would you figure out the unknown? So this part of the data, you are using it to construct a standard curve. Okay. Using that standard curve, how do you figure out what is the concentration of the unknown sample? Okay. You will have to figure out the concentration of the unknown sample, which is what you did for the enzyme report. So you have already done that, right?
Yes.
Yeah,
Mhm.
okay.
Yes.
So let me extend this, okay? So this is how you do the standard curve, okay? So you have all these different concentration. Okay. So you have different concentration of color, different intensity of color. You read the absorbance, and then you construct it against the absorbance. As you increase the concentration, you get increased color intensity, okay? Based on that, you make a standard curve. This is what you did for your maltose assay, right? For your maltose assay, this is what you got. This is what your tubes look like, right? That you had an increase in color, right?
Yes.
Using the standard curve, this kind of graph, your unknown will have an OD, okay? You do the same experiment, one of the tubes will have your unknown, and you will read its absorbance. Draw a line that intersects your standard curve. This is the same standard curve. Draw a line which will intersect it, and then you can read it off of this, okay? So this is one way of getting your concentration. You may be given an equation for the standard curve, okay? You may be given an equation for the standard curve, and you can use that to figure out what is the concentration. Okay, let me see if I can find one. Nope, hold on. Not letting me paste it. Nope, it's not letting me paste it, but you have done this before, right? Okay, so I guess not. But you have done this before, and you know that if you know the absorbance of the unknown, you can read it off of the standard curve. That's the whole point of doing the standard curve, so that you can use any of these absorbances within the range and read it off of this. If you have the standard curve, then you can get the equation for that standard curve. I don't have, it's not letting me copy it. Okay, so can you see this one?
Yes.
Okay, so here is a standard curve, and let me do it here. Okay, so here is the standard curve, and this is the equation. If you are given the equation, you should be able to calculate what the concentration of maltose is, knowing what the Y is. Your Y is the absorbance of your unknown. You plug in the absorbance for the unknown, and X is going to be what you are solving for. That's what you want. Okay?
Mm-hmm.
It's giving me, no, for whatever reason, it's not letting me do it on whiteboard. Okay. All right. So make sure that you can do that. You can use the equation to figure out what is the concentration of the unknown. Okay. Okay. Let's get into the bioinformatics part and then we will take a break. We have a long way to go. And Tyler was right. I don't know if Tyler is still here, but yeah.
Yeah,
Okay.
he's right, Professor.
Oh, are you still here?
No, no, I'm not Tyler. I'm another person.
okay.
I am still here.
Oh, you are still here. Okay. Yeah, that was a good pointer. Thank you for that. I'm looking, I always get carried away with making sure that everybody's comfortable. Okay, so here we go. We are going to go into bioinformatics part, okay, which is we're looking at the structure of proteins. So this is a structural and functional analysis of the enzyme. So enzymes are complicated because one, that they are made of proteins. So you have to not only look at the structure of it, but also its activity, because there is a reason why we are using this protein, because we can measure its activity. It is doing something that can be analyzed, okay? So that makes it a very interesting lab, very exciting lab, because you can do all of that, okay? I'm going to move into the bioinformatics part. And it's a shame that the videos are not working, because this was let's see if it's, maybe even if you cannot see it, hear it, if you cannot hear it, it would still, so I can kind of walk you through, and there are what do you call it? It's written here, so you should be able to see it also.
Yeah, we can see the videos, but we cannot hear them. That's the thing.
Yeah.
Yeah,
So,
but we can read because whatever
right,
is
right.
the
So you
explanation.
can read it. So I'm glad that it actually has that. I'm going to put the link in the chat so you can save it. Okay, so you can save it and hear it later on, but you can read it here. Okay.
Professor, sometimes,
Sorry.
yeah, sometimes I hear in the background look like it's the video sound, but not sure what is. But that's okay. We can read.
Okay.
We can
okay.
see the video.
Yes.
That's
Watch
right.
the video and you can read it
Yeah.
here. Okay. So it's transcribed. So the reason I want you to see this is a very good one that kind of visually shows you. So it's worth the time. So the reason I wanted you to see this is all of these shapes, the secondary structure, the alpha helix, the beta sheet, the flat arrow, the beta sheet, these are part of its tertiary structure, okay? And these are important. These are mediated by what amino acid is making up that part. Some of it does not have a secondary structure, okay? Some of these have these bonds to which the substrate is actually binding to the enzyme, chemically binding to the enzyme. And that depends on what that amino acid is in that position, okay? So when we look at all the amino acids, the 20 different amino acids that make up the polypeptide chain, that is the primary structure. To maintain the 3D structure, it needs to go through the alpha helix, the beta sheet, which are examples of the secondary structure mediated by hydrogen bonds. And then once it is folded, it then further folds into the tertiary shape, okay, to have the overall 3D shape, okay? Now, bioinformatics is the analysis of the different kinds of amino acids that make up that polypeptide. So we are using the alpha amylase. We use 3 different kinds of amylase in the lab. And so this is the computer analysis using computer software and different kinds of computational tools to analyze what amino acids are there, what kind of amino acids are there, because there are 20 different amino acids. And if you actually look at them, each amino acid is not just structurally different, the chemical properties are different. Some are polar, some are non-polar, some are acidic, some are basic, some are hydrophobic. So there are many different kinds of amino acids. And so depending on where that amino acid is, it will fold, it will direct how the polypeptide chain is going to fold. Is it going to form alpha helix, beta sheet, hydrogen bonds, disulfide bridges, whatever folding it's going to have, that will depend on what amino acid is there, okay? Now, when we analyze the structure, we can learn about it, and that will explain why some of the amylases have such high activity at 85 degrees, and some of them get denatured at 85 degrees, okay? That was the purpose of doing this analysis and learn some of the bioinformatic tools, okay? For your exam, you will be given enzyme ID numbers. See these accession numbers? You will be given these, and the sites will be there. You don't have to know what the sites are, but you will have to go to the site and figure out what to do at that site, okay? So one of the things you need to know, the different links that we used. So NCBI was used to just get the amino acid sequence, okay? Get the amino acid sequence for all the different amylases, okay? So one of the things to remember is that when we get to the site, and let's say, so this is a hyperlink, I'm just gonna go there. And you are going to get to this chain. It will, this page, this is the format, that's the default format at NCBI, the National Library of Medicine. This is the NCBI is the biotechnology information. If you scroll all the way down, this is where the amino acid sequence is. Okay, this is the protein, this is where the amino acid sequence is. But notice the numbers here and the gaps here. This is going to not work with a different tool that we are going to use next. Okay, this is just a sequence. It's not really useful in that sense. It just tells us how many amino acids are there. And it tells us there are 496 amino acids, okay? But to be analyzed what kind of amino acids are there, how many are polar, how many are non-polar, we will need to obtain a format, the sequence in a format that doesn't have those spaces and numbers. And that is the FASTA format, okay? So you can go here, or you can get it from here, get the FASTA format from here, okay? So if you go here, and you can copy the whole thing, and this is something you have done, okay? And I'm going to delete that. Oops. Okay, so this is the FASTA format. And the one that we just took was, which one? It was the human amylase, okay? And so what you would do is write down human amylase, okay? And this is the FASTA format. This is the format that works with most of the software, most of the tools that we use for analysis, okay?
So a question for the exam, how this question is going to be?
Okay, so the question is going to give you accession number, which is like the MCID that you all have.
Yes.
Okay, it's going to give you the accession number and it will give you the site.
Correct.
Okay, and so what you need to do from that site is get the FASTA format, the amino acid sequence. Okay, that's all you can do from the NCBI set. Okay, and then you are then going to analyze the primary structures, going to PepStat. Okay, that will also be given to you, but you need to know, what do I do here and what do I do here? What is the purpose of all of this? Okay, that's what you need to know. So the reason I copied that is because you are going to find out which of the amino acids. So here, all the amino acid sequence that I copied here, which of these amino acid is polar, which of it is non-polar, how many amino acids are there. You could sit there and count the whole thing, but there are softwares which will do it for you. That's the purpose of it, okay? And so
will we be having like a computer to look
yes,
at? Is it
yes.
like
At the station, you will have laptops. Yes.
Oh, I see.
Okay.
Okay. You will, for that station, for the bioinformatics station, you will have laptops where you will have the sequence already there that you can copy and paste and all of that. Okay. And you will get the instructions in your question.
And will the laptop be like the school's laptop or
Yes.
your own?
Yes. No. The laptops will be at the station. So no, you cannot use your phone. You cannot use your own device. Nothing. Okay. So this would be the site where you would be going, and let's see, you paste and just submit it. Okay. And you should get a screen probably within a couple of minutes. Typically, you would get, is it going to do it? Okay, here. Okay. So this is what it's going to look like. Residues means the number of amino acids. Okay. So it's 496 amino acids. Okay. These are the abbreviation. You don't need to worry about that. Just go all the way down and find out this one, the non-polar and the polar ones. Okay. So it tells you how many of those 496 are non-polar and how many are polar. So 270 amino acids are non-polar, 225 are polar, and that's the percentage. Okay. This is all you need to find out.
So they will ask a specific question, the numbers of say non-polar or polar, or also the percent.
So this is the number, this is the percentage.
Okay.
Okay, so whatever the question is asking for, you know that this is the number and this is the percentage, okay?
Professor, I'm sorry to bother. Do you mind to kind of go back when you submit this? I just missed this. How did you?
Okay, so again, I'm going to quote Tyler here that it is being recorded, okay?
Oh, okay.
The session is being recorded and you will have access to the recording, okay?
Yeah, okay.
Okay. So this is where you get the non-polar and the polar amino acids. So that's what you are doing at the PepStat. Okay, so some of the questions will be like, whatever accession number is, it doesn't have to be amylase. Any accession number that is given to you, you can go there and find out how many of those amino acids are polar, non-polar, what percentage, all of that. So it does not even have to be amylase. You can do it for any protein. Okay?
Okay.
Yes.
All right. So don't expect that it's going to be amylases. It could be anything. okay? But just know how to use it. And what is the purpose of NCBI? To get the FASTA format, the amino acid sequence in the FASTA format, and then go to the PepStat to do the computation, to figure out the chemical properties of individual amino acids. This tool can provide information about the chemical nature of amino acids, okay? So the other one that you should also practice doing is going to the PDB site, which is the protein database, okay? That tells you what percentage is the secondary structure motif, what percentage is alpha helix, what percentage of the secondary structure is beta sheet, all of that, okay? Because that has to do with the overall stability of the protein at high temperatures, okay? Typically, if the protein is thermostable, meaning that it can withstand high temperature, it has more of the beta sheet than alpha helix, because beta sheet is more rigid. It does not fall apart that easily. So enzymes that work better at high temperature, like this one, it will have more of the beta sheet than alpha helix. These ones, human and fungal amylases, these ones will have more of the alpha helix than beta sheet because it offers flexibility. These are coiled. Alpha helix is spiral, coiled like that. So it has more of those alpha helices, okay? And you will find it from the protein database, okay? So this is the PDB ID. You are going to use that. Let's see, do I have it here? Up here, okay, yes. So here, this is the site where you are going to go. And again, the same thing, you will be given accession number. You are probably going to have to copy it and go there, okay, to get to the site and find out what is the percentage. So here, I think we had it here, okay? So percent of alpha helix in human, 27% is alpha helix, 23% is beta sheet. Look at fungal, 34 is alpha helix, 21 is beta sheet. But if you look at bacterial, 30% is beta sheet, because the bacterial amylase, anybody use bacterial amylase in your exam, in your experiment?
I don't think so.
Nobody in your class? Anybody here use the bacterial amylase?
No, we did the fungal.
You use fungal. Nobody used bacterial. None of you did?
I did.
You did. Do you remember what was the optimal temperature for your experiment? Do you remember?
I think it was between 55 and 65.
It should be around 80 degrees.
Around.
Yeah. Okay.
Oh.
So one of the temperature that we had used was
You mean
85.
the optimal, right?
Optimal, yes.
Oh yeah, 85. I'm sorry. I
Yeah.
keep thinking about
So the bacterial is optimal, meaning the highest activity is at 85, where most of the enzymes would be completely denatured, or you would have the graph would be very, very different. Okay. So where it drops for human and fungal amylase, bacterial amylase is actually increasing in activity around that temperature. Okay. Because it is from a hyperthermophile, a temperature where a very high temperature where these bacteria are surviving. Okay. And it's taken from those bacteria. That's why it has that doesn't mean that all bacteria have it like that. It just happens to be that particular one. Okay. Okay. So coming back again, the screenshots are here that will tell you again to what it should look like. It may look different because these screenshots are from pre-pandemic time. And so in bioinformatics, they keep on changing. We found out that one of the tools that we used to use does not exist anymore, not available anymore. So we had to use something else for that. So the screenshot may look slightly different I don't think so this semester. These were fine. These were the same. How can you obtain FASTA sequence and why do we need FASTA sequence? You can obtain FASTA sequence from NCBI site, okay? Why do we need it? Because it is compatible with the other software that we need to use to analyze the proteins, the amino acid sequence, and other computational tools, statistical tools, okay? So that's why we need the FASTA format, okay? So if you want to know how many amino acids, how many polar, non-polar, all of that, okay? Okay, I think for the bioinformatics part, you should be good. I'm going to skip this one, okay? This is kind of telling you about what these three different enzymes are. If you look at compare the three different enzymes, human, fungal, and bacterial, substrate is the same, product is the same, okay? And if you look at the amino acids that are at the active site, these are the three amino acids that are directly involved in binding to the substrate, okay? Even though the amino acid sequence similarity between human amylase, fungal amylase, and bacterial amylase, if you look at that, what percentage of the amino acids are similar? 20 to 30%, 20 to 30%. So, which means 80 to 70% is different, okay? And yet, they are converting the same substrate into the same product, okay? And the reason they are able to do that is because the amino acids at the active site that bind to the substrate are conserved for all three of them, okay? Even the number of amino acids are different. Rest of the amino acids are different. The overall shape may be different. Their optimal temperature is different. Optimal pH is different. And yet they are converting the same substrate into the same product, okay? So even with all of these differences, the reason they are able to do that is because the amino acids that are directly involved in binding to the substrate is the same. That did not change, okay?
Yes.
Yeah, okay.
Yeah.
I'm going to pause here and let's see. And let's take a 10-minute break. I'm gonna get some coffee and we'll be right back, okay?
Okay, thank you so much.
All right. Okay. Okay, so we are going to get started and we'll start with exercise 10, which is the genetics. This was the last lab that you have done. And some may still be doing the assignment for it, but this is the one that was 2 days, another 2-day lab where you started with part of the experiment and then you continued with restriction digestion on day 2, right? So this lab was about analyzing an inherited disorder, sickle cell disease in a hypothetical family and hypothetical parents with 2 kids, and you were analyzing their DNA to find out if any of the kids are have the trait for sickle cell or any of them have the disease. And so that you had to do the, I think can you hear me?
Yes, we can hear you.
Yes.
Oh, okay. I think my AirPods just made that sound that kind of sounded like it didn't have enough power. Okay. So what you need to understand is we have been talking about sickle cell disease, what the disease is, and what causes that disease, and how is it an inherited disorder, okay? It is a genetic disorder where the hemoglobin that carries oxygen to the cells, the hemoglobin is made up of 2 subunits of alpha chain, 2 beta subunits, okay? So 2 alpha subunit and 2 beta chains, 4 of these come together, have a quaternary structure to make 1 molecule of hemoglobin. Okay, so each hemoglobin molecule is made up of 4 polypeptide chains, 2 alpha subunit and 2 beta subunit. The disease is caused by a mutation that affects only the beta chain. Okay, so the mutation is in the HBB gene that has a, does everybody know what mutation means? What does the word mutation mean?
Like I've heard a lot of it, like in terms of viruses and bacteria, but I don't remember the definition of it. I mean, the exact definition.
Okay, so mutation means that any change in the DNA. Okay, any change in the DNA. DNA is made up of nucleotides. It's a polymer of nucleotides. And if any of the nucleotide changes, that is a mutation. Okay, and this change can happen for many reasons. Some of the changes are stable, some of the changes are not stable, but some of these changes that cause a disease, depending on where that change is, it may or may not even affect. Doesn't always mean that because of that change, there will be a disorder, okay? But this is a classic example where only one nucleotide has changed and that results in a disorder, okay? I have, in the chat, I have posted a whole bunch of, oh, actually, you know what? I think I have sent it to, I thought I was sending it to everybody, but I guess not. Okay, I have posted a whole bunch of YouTube links, but I posted it to the last person who sent me a message, okay? So the videos that I am talking about, let me post it again. So since you can't hear me,
We can hear you.
sorry, I meant like hear the YouTube videos. That's what I meant, sorry.
I'm sorry.
Hold on. I'm gonna post it again.
Oh, I think we have a visitor, Professor.
You have,
No, we have a visitor here.
we have a visitor?
Yeah.
What do you mean a visitor?
There's a child, I think. I see a child.
Okay, well, somebody is interested in the review then. Okay. Okay. So I am posting some of these videos. Okay, you can see it. Okay, good. Thank you. So here is another one on polymerase chain reaction. And this is the one on RFLP, which is what we are using. We used in the lab, okay? And I'm going to go through all of these, but these are nice videos and animations that I have used for my class and I have provided for my classes. So and the last one here. So these videos are the ones that I was planning on using, but guess not. Can you see all of the links? I think 5 of them that I posted. Can you guys see it? So save it on your device. You can watch it later. Okay. So the gene, let me get back to the whiteboard. So sickle cell anemia, the gene that has the mutation is the gene that codes for the beta subunit of hemoglobin. Okay. Hemoglobin is made up of 4 polypeptide chains, 2 alpha subunit and 2 beta subunit. The mutation is in the gene that codes for the beta subunit. Okay. And only one nucleotide changes that causes the disorder, sickle cell anemia. Okay. Now, the gene for beta subunit is located on the chromosome in number 11. Okay. So can you see the screen, the whiteboard?
No.
No, yeah.
No? Okay, hold on. How about now?
Yes.
You can, okay. So the gene for beta subunit of hemoglobin is located on chromosome number 11. You know, humans have how many chromosomes?
46.
46, 2 sets of 23 chromosomes, right? And so the gene for beta subunit is located, the allele for beta subunit is located on chromosome number 11. Our cells are diploid cells, right? So we have 2 chromosomes, 1 paternal chromosome and 1 maternal chromosome. And this is the locus, this is the position, HBA. This is the position for the gene for beta subunit, okay? So these are the genotype. If a person is normal, does not have the mutation, does not carry the allele, this is the gene for the HBA is the normal allele, okay? So the genotype for normal person would be HBA/HBA, meaning both the chromosomes are normal, okay? So this person does not have the mutation. This individual, the paternal chromosome has the mutation. The maternal chromosome does not have the mutation. So it is a heterozygote. This individual is a heterozygote and will be a carrier. So can pass on this mutation in the beta subunit to the offspring. So this person will not have the disease, but will be a carrier, okay? The individual with the sickle cell, it is an autosomal recessive disorder, meaning that the individual, in order for the individual to have the disease, has to have both the alleles that have the mutation, okay? So it has to be a homozygous recessive with the mutation, okay? And so that individual is going to have the sickle cell trait. So these are the genotypes. This is the phenotype, okay? Now, the HBV allele, the gene, is 1,608 base pair long in chromosome number 11. So this is kind of showing you the DNA. So this would be the DNA. This is made up of 1,608 base pair double-stranded DNA, okay? The gene starts from here all the way up to here, okay? And that's the 1,608 base pair. For eukaryotes, for humans, for all eukaryotic cells, the genes have these coding segments and non-coding segments. The exons are the coding region, okay? Introns are the non-coding region, okay? The reason I'm going through this is because you had to find these for your assignment, okay? So these exons, there are 3 exons. This is what was given to you. This is what was given to you, right? The 3 different exons, okay? So this is 1 exon, this is 1 exon, and this is another exon. All of this sequence in between, that is the intron, okay? That is the intron.
Excuse me, Professor, can you clarify what is the exons there? The one that
exon is the coding region. That's the one that codes for proteins. Introns are the non-coding region, okay?
Yeah, no, I mean here in this table.
In the figure? In the figure, all the ones in the highlight are the exons. The region in the highlights are the exons. The non-highlighted portions are the introns.
Oh, I see. Thank you.
Okay. So when the gene is transcribed, it will first make a pre-mRNA. Okay. This is the pre-mRNA. It's going to make a pre-mRNA, which is actually pretty long because it will copy the introns. The whole thing is copied. Okay. So it will make a pre-mRNA, which is pretty long, and then it is going to go through. So when it is first transcribed, it will have to go through processing. Okay, the mRNA is very long and it will go through processing. The processing involves removal of the introns. So these parts that are the non-coding part, these will have to be cut out. Okay, they have to be removed. It gets a 5' cap, a 3' tail. Okay, these are part of the processing. And so it ends up making a much shorter piece of mRNA. This is the mature mRNA that goes and binds to the ribosome to make the beta globin. Okay, now notice in the mature mRNA, this black region is not there, right? Because this is cut out. These are all cut out, okay? These are the introns, these are the non-coding segment, and these are removed. And so just the part that is the coding segment are stitched back together, okay? That is called RNA splicing. You don't need to know that for now, but just know that the non-coding segments are removed, gets a 5' CAG and a poly A tail, and the piece, the gene is this long. After processing, it is almost 1/3 of the size, okay? This then goes to the ribosome and makes the protein, which is translation. So this is the protein. Beta globin is the protein made up of 147 amino acids. Okay, so it gets folded into the 3D shape and is 147 amino acid. Okay, so this right here is the mRNA. This right here is the mRNA, and the part in the green is the protein part. Okay, this is the part that gets folded and becomes the beta globin. Okay, that's the start codon, this is the stop codon, and this is where the amino acids are added. This right here is the actual amino acid sequence, okay, made up of 147 amino acids. Okay, this is the amino acid sequence of the beta globin. Okay, now what you had done for your experiment, you took that RNA and amplified it. You had a DNA, which is the same as the mRNA. mRNA was used to convert into DNA. And what you were given was DNA from these individuals, okay, from a family. And you amplified that specific DNA from the beta globin, a small part, okay, about 233 base pairs. So from the 628 base pair DNA, you amplified 233 base pair, okay? That's the part. So if the entire DNA is this long, you amplified a 233 base pair. So this is the amplified, meaning making a lot more of it. Why? So that you can analyze the DNA and find out if that individual is heterozygote, if the individual is homozygous, is the individual carrier, you can find that out, okay? This is done by what is called restriction fragment analysis. But before we went to that on day one, what you had done is a reaction to amplify the DNA, which is called PCR. Anybody know what that stands for?
Well, I remember for PCR is like well the most famous example can be like when you get a test for COVID-19.
Mm-hmm.
And another example of PCR is like when the PCR makes like replications of DNA as well. Is related to the DNA replication, I think.
That is correct. It uses DNA as a template, okay, and makes more DNA, okay? This can happen in the cell when the cells are dividing. It is making copy of the entire DNA. That is called the DNA replication, okay? DNA replication that happens in the cell uses the same enzyme, DNA polymerase, but it will copy the entire genome. Polymerase chain reaction is also copying the DNA, but you are doing it artificially in a tube, and you are not copying the entire genome, okay? You are copying a segment, a small segment of the DNA and making many, many copies of that specific DNA, okay? So not the entire genome, but just a segment, a selective region of the DNA is amplified, okay? Kind of think of it, and you can control as many as you want. When DNA is replicated in a cell, if this is a cell and DNA replication is going on, then it will start with one DNA molecule and make two DNA molecules, okay? So you just get two copies of the DNA. But in a PCR reaction, you can get as many copies as you want, depending on how many times you repeat the PCR reaction, okay? So even if you cannot hear it, I want to kind of show you the video just because that will explain what is happening in the PCR reaction. In a tube, what you do is you add a number of reagents. One, that you add template DNA, whatever DNA that you want to make copies of, okay? So template DNA, which was coming from the family members, okay? You had to add the DNTPs, which are the building blocks, okay? Or nucleotides. DNA is made up of nucleotides, okay? So nucleotides are there. There are primers that are added, 2 primers, forward and reverse primers. There is buffer. And most importantly, there is a Taq polymerase, which is a heat-stable DNA polymerase. Okay, for your PCR reaction, when you did it in the lab, all of this was already added in a reaction mix. Okay, you didn't have to, you had a reaction mix. All of this was already added for you. All you had to do is add the template DNA from each individual. So you had set up PCR reaction in 4 tubes, okay, where the only difference was the template DNA that was coming from each of the individuals. And you were given a reaction mix, which had all of these already added, okay? Now, how does it do the reaction? That's what I want to show you in this video. Okay.
The polymerase chain reaction, or PCR, is a technique that can produce over a million-fold amplification of target DNA within a few hours. PCR requires specific oligonucleotide primers and plenty of nucleotides for DNA polymerization. It also requires a heat-stable DNA polymerase because PCR requires repeated cycles of heating the solution to 95 degrees Celsius.
Okay, so see these primers and the nucleotides that were already in the tube. Okay, these were already, so these nucleotides are the building blocks. What is DNA made of?
Proteins and histones? Sorry, no. That's
Proteins and DNA.
my bad.
Nucleotides.
Nucleotides.
Nucleotides, yes. DNA and RNA. DNA and RNA both are nucleic acids, right? Are nucleic acids. Nucleic acids are polymers of nucleotides, which means nucleotides are the building blocks. So if you want to make DNA, whether you want to do it in the cell, whether the cells are doing it, or we are doing it in a tube, okay? And when it's happening in the cell, we call it in vivo, okay? When it is happening in the tube, we can do this in a tube. It's called in vitro, okay? Outside of the cell, in vitro. When we are carrying out the DNA replication in a tube, we can do the same thing, but we are going to need the nucleotides, okay? So in the PCR tube, we provide all the nucleotides. It also needs an RNA primer. It needs RNA primer when it is happening in the cell, but in the tube, we use DNA primers, okay? We still need a buffer. We need a DNA polymerase, but in the tube, we use this Taq polymerase. In the cell, it has its own DNA polymerase, okay?
Celsius. The polymerase Taq from the thermophile Thermus aquaticus is often used for this purpose. In PCR, the reaction mixture is heated to 95 degrees Celsius for 30 seconds to denature or separate the DNA strands.
So there are 3 reactions that are repeated over and over in each of the cycle, okay? The first step is called denaturation, which is separation of the double-stranded DNA, and that happens at 95 degrees, okay?
The temperature is then lowered to 55 degrees Celsius for 30 seconds, which allows the oligonucleotide primers to anneal to known DNA sequences.
So the next step is called annealing, okay? The second step is called annealing, which is carried out at a lower temperature. Annealing is the primers that were in the tube that allows these primers to bind to the single-stranded DNA, okay? So this part of the DNA that has become single-stranded, the primers anneal to it, and that provide a 3' hydroxyl group, a free 3' hydroxyl group.
Flanking the target DNA. The oligonucleotide primers, which are usually 20 to 30 nucleotides in length, are made so that they're complementary to the flanking sequences. The temperature is then brought to 72 degrees Celsius, which is the optimal reaction temperature for Taq DNA polymerase.
So notice the 72 degree here. This is the enzyme, the Taq polymerase, which is the thermostable DNA polymerase. That's the optimal temperature for this enzyme. And this then starts to add complementary nucleotides at the 3' end of the primer and keeps going, okay? So this step is called extension, okay? The 3 steps of each cycle of PCR, the first step is denaturation, very specific temperature, denaturation at 95. That's step 1. Step 2 is annealing, where the primers, these primers would anneal. Denaturation would separate the template DNA, annealing at 55 degrees. And then extension of the primers by adding complementary nucleotide at 72 degrees, okay? These 3 steps per cycle, okay, are repeated over and over, okay? Until we get as many DNA as we want. And we can determine how many cycles we want. Kind of think of it the way, let's say I have a book, and here I have a book, and I want only 2 of these pages, page 93 and 94, I want 200 pages, copies of those 2 pages. So what I would do is I would put this on the photocopier, set it to 200, and just copy these pages. So the entire book is not copied, but only those 2 pages, and I can set as many number as I want, right? PCR is kind of like that it would copy a specific segment of the DNA as much as I want by setting how many cycles I want, okay? But if it was DNA replication happening in the cell, it would make a copy of the entire thing, and it would make just 2 copies, okay?
The polymerase uses the primers as starting points for DNA synthesis, adding 1 nucleotide at a time to create a complementary strand of DNA.
These are the building blocks,
The cycle
right?
is repeated, starting with the 95 degrees Celsius heating step, cooling the solution to 55 degrees Celsius.
So this is the second cycle, okay? This is the second cycle, and it will be repeated.
Allows the primers to anneal. Heating the solution back to 72 degrees Celsius allows polymerization to proceed. After 2 cycles, 4 double-stranded target DNA sequences exist.
So 4 copies, 2
After
cycles.
the 3rd cycle, 8 copies exist. After the 4th cycle, 16 copies exist. After 25 cycles, about 30 million copies of the target DNA exist. Because
So if you wanted to find out how many molecules of DNA would be there. You can do the calculation with that formula 2 to the power n, okay? So 2 to the power n, where n is the number of cycles. N is the number of cycles, okay? So we can program the thermocycler to make as much DNA as we want, okay? So this is the technique that was used to amplify the gene. This was used to amplify the DNA for this specific size of the fragment, okay? So we amplified a region of the beta globin gene so that we get this size of a fragment. And the reason we wanted this segment is because this includes a site for a restriction enzyme. Anybody know what restriction enzymes are? Anybody know what restriction enzymes are?
Not by heart.
Okay, so restriction enzymes or are enzymes, of course, so they catalyze a reaction, okay? This is how we write it. These are called nucleases, okay? Another name for that is nucleases, meaning that they hydrolyze and cut DNA.
Oh, they
okay?
cut, yeah. Okay.
They cut DNA. So nucleases, their substrates are nucleic acids. So DNA and RNA are nucleic acids, okay? So their substrates are nucleic acid, and they would cut the DNA into fragments, into pieces of DNA, okay? So kind of think of it as a pair of scissors, okay? That if you have DNA, it's going to cut the DNA into pieces. So if you have a double-stranded DNA, and this is one of the enzymes, what this is going to do is it's going to cut it right here, okay? So it's going to cut it like that. So if you have DNA like this, the enzyme is going to make a cut like that and break it up into pieces, okay? But they are restriction enzymes, meaning that they're not going to randomly cut DNA anywhere, okay? They look for specific sites that is called recognition sequence. Okay, that is called recognition sequence. They scan the DNA, look for that site, and that's where they are going to cut, okay? For the DDE1, the recognition sequence is between A and T. So here, I'm going to take you back to, okay, so here, so this CTGAG, this in the box, see that asterisk? That is where, this is one strand of the beta globin gene, okay? This is where the enzyme DDE1 is going to cut, okay? The PCR product that we amplified is up to here. So from here, this is the end of that, and this is the start of that. So it starts from here and ends up to here. If you actually count it, this would be equal to 233 bases, okay? So starting
Professor,
here.
a question. So we always gonna to identify what is gonna to be cut because it's in that box?
No, it depends on the enzyme. So you need to know what restriction enzymes are. You don't have to memorize it. If you are getting the sequence, you should be able to tell where it's going to get cut. Let me show you in your lab manual. So this is where it's talking about the endonucleases, what restriction enzymes are, or restriction endonucleases, and the recognition sequences, okay? So these are the names of different enzymes, EcoR1, DDE1, BAMH1. These are all different kinds of enzymes, okay?
Mhm.
And so see where the arrow is, that means the bond between G and A. This is the recognition sequence for EcoR1, GAATTC, going from 5' to 3' and 3' to 5', the complementary strand. This particular one is the 6 base pair. So 1, 2, 3, 4, 5, and 6. 6 base pair recognition sequence. The enzyme is going to bind to the DNA and look for its recognition sequence. Once it finds it, it is going to cut one strand between G and A and the complementary strand between G and A at this arrow, okay?
I see.
And it is going to make fragments like this. The ends where this part right here is sticking out, this part right here is single-stranded. These are called sticky ends, okay? These type of ends are called sticky ends. Some enzymes cut in a way that it produces these sticky ends. Some enzymes cut right in the middle and do not produce any sticky ends. Okay, those are called blunt ends. So the two different kinds of ends that the enzymes can make, enzymes when they cut, one is called sticky ends, okay? And the other is called blunt ends, okay? And when you look, when the sequence is given to you, you should be able to tell whether it's going to make blunt ends or sticky ends, okay? Because there will be an arrow pointing where it's going to cut, okay? So if it produces ends like this, then it is sticky ends. The blunt ends will look like this. There will be no single-stranded part, okay?
Okay.
That's what the blunt ends are going to look like. So this is also going to produce sticky ends, okay? Now, some sequences that are symmetrical, for example, this one, if you read from the 5' to 3' end, it reads GAATTC. If you read the bottom strand from 5' end to 3' end, GAATTC, it's reading the same, right? These type of symmetrical sequences are called palindromes, okay? These type of symmetrical sequences are called palindromes. And so you should be able to look and read it from both the strands from the same end, 5' to 3'. And if it reads the same, that means it's a palindromic sequence. Like the words mom or wow, where you can read it from either side and it reads the same, right? Those type of sequences are called palindromes, okay? So when it makes the fragments, how do we use those fragments? So one, that we use PCR to amplify a specific segment of a gene that carries the site for this restriction site for this enzyme, because when somebody has a mutation and has the sickle cell anemia, within that site, that recognition sequence is changed, okay? One amino acid changes, and that would be this A, okay? This A changes, okay? So if the A changes, the enzyme DDE1 does not recognize it anymore. And so it's not going to cut it because that A has changed, okay? Because of that mutation, the A has changed, and going back here, okay? So see this one right here? This is what you have in your exercise. This is the start codon. This is where the primers bind, the red ones. This is where the primers bind. This is the start codon. This is the stop codon. And this is where the restriction site is, okay? The PCR product starts from here all the way up to here, okay? So the part that I highlighted, this is what it's going to amplify with the primers that we have used. This is where the forward primer is, and this is where the reverse primer is. It is only going to amplify this much of the gene, okay? And that is equal to 233 base pair. That's the size. The size of the DNA, the units that are used are in base pair because DNA is double-stranded. So there are two-stranded. Here it's showing you one of the strands. It's not showing you two strands, okay? It is a double-stranded molecule. And for simplicity, we oftentimes show the single strand, but here I have the double strands. Okay, so see that two strands, the complementary two strands.
Mm-hmm.
So the size of the DNA, because DNA is a double-stranded molecule,
Mm-hmm.
okay, because it's a double-stranded molecule, we always write the size of DNA as base pair because the bases are complementary bases are paired. Okay, if it was single-stranded, like the RNA molecule is single-stranded, we would write it as pair, as bases, not
Bases.
pair, because RNA is single-stranded. Okay. Okay, so I am going to go to, okay, if you guys cannot hear me, let me know, okay? I'm getting these weird noises that kind of tell me something is powering off, okay?
Maybe it's your headphones, Professor. I mean,
Yeah, I think it's draining charge or something's happening, but as long as you can hear me, that's fine. If you cannot hear me, let me know, okay?
yeah. Yeah. I mean, maybe just try and charge your headphones later.
Yeah, I think I may have to switch to my headphones, okay? All right, so here, the exon that I was showing you in that handwritten diagram, exon 1, 2, and 3, and the DDE one side, this is in your exercise, that's what I'm showing you here, okay? So the exons are in yellow, this didn't come up with yellow, but what I have highlighted in blue is the part that we were amplifying by PCR. We made many, many copies, okay? We did a 30 cycle, okay? Which means you should be able to tell how many copies of this segment was made.
Mm-hmm.
How would you figure that out? 2 to the power 30, okay? Where 30 is the number of cycles, number of PCR cycles, okay? So you would have this many DNA molecules, okay? So going back to page 8 in your handout.
I'm sorry, how many DNA molecules was that again?
2 to the power 30, because that's how many times we repeated the PCR cycle.
Okay.
Somewhere here, it was here. See, after 30 cycles of PCR. Okay?
Okay.
So you should be able to do this. If we have one cycle, you would have 2 copies. Okay? To do 2 cycles, 2 to the power 2 is 4. 2 to the power 3 is 8, right? So you should be able to fill all of that. 2 to the power 4 would be 16, right? So every time the number of DNA molecules double. Cycles would be 2 to the power 30. Okay? Okay. Can you still hear me?
Yeah, we can.
Okay, so now my AirPods completely dead. Let me see if I can switch to headphones. Give me 1 second. Okay, can you hear me?
Yes.
Okay. All right, good. Okay, so going back to the number of cycles and how many copies would you get, okay? So this is where you would find it, page 10, 11 is where you would find it, okay? So I'm going back to the handout. What are the main steps of a polymerase chain reaction? Briefly describe what happens during each one. And that's what I was talking about here when I was showing the 3 different steps: denaturation, annealing, and extension. Denaturation, the double strands are separated. Annealing is when the primers anneal to the single-stranded template DNA. And extension is when the primers are extended by the enzyme Taq polymerase at 72 degrees. Okay, so those are the 3 steps. The PCR reactions happens at a very high temperature. How come Taq polymerase is not denatured? The reason Taq polymerase is not denatured is because it is taken, the enzyme DNA polymerase is taken from a thermostable organism. Okay, Thermus aquaticus, that's the name of the organism. And while the video will also give you the name, Thermus aquaticus. Okay, that's the name of the organism. That's where the name comes from. Taq polymerase, Thermus aquaticus. That's where the name comes from. Okay. And you will find the name here also in your exercise. It will talk about where the enzyme is coming from, the thermostable enzyme, okay, that is used, and it comes from Thermus aquaticus. That's the name of the organism. That's where it gets the name Taq, okay? So this is on page 3. Okay, what are the main differences between the steps of DNA replication and the steps of PCR? One of the major difference is the enzyme DNA polymerase that is used. The enzyme DNA polymerase that is used in the cell is its native DNA polymerase, okay? And it will only double the number of copies, okay?
Excuse me, you say it's because it's what? Negative? No.
Native, that it's cell's own, only
Okay.
its own DNA polymerase, okay? For PCR, it is the Taq polymerase that is used, okay? In DNA replication, the primer that is used is the RNA primer, okay? In PCR, it's a DNA primer that is used, okay? The reaction that takes place in PCR, the temperatures are very different. Each of the steps is carried out at a different temperature. In DNA replication, all of it is the same temperature. There is no variation in the temperature, okay? So one of the questions that you had here where you had to compare the processes in your assignment was to compare in the PCR assignment, you had to compare what is the difference. Was it here or is it in the assignment, I think? Hold on.
That's
Sorry, I'm scrolling way too fast, but I'm trying to find that question where you had to figure out here. Yes, you have it here. So DNA, this is the question that it is referring to. What is the template that is being copied? In E. coli DNA replication, it's the chromosomal DNA, okay? In PCR, we are copying the HVA gene, okay? HVA gene, a specific part of the HVA gene. How many copies of the template will result? Here you will have only 2, and here whatever many we want, okay? In this case, it was 2 to the power 30. What bonds, what separates hydrogen bonds from DNA strands during replication? So in this one, it's the enzyme, okay? The enzyme helicase is going to separate the DNA strands, and here 95-degree temperature breaks the hydrogen bonds to separate the DNA strands, okay? Here, what provides the 3' hydroxyl group for extension? Here, this will be the RNA primer. Here, it will be the DNA primer. Okay, so here is the RNA primer. Here would be the DNA primer. What is the name of the DNA polymerase that is used for extension? That will be the DNA polymerase. Here would be the Taq polymerase. The optimal temperature for DNA polymerase, this would be 37 degrees. This would be 72 degrees. Okay, so there are lots of differences. That's what this question is asking for. What are the differences in the DNA replication? Okay. How many DNA copies would theoretically be made if you allowed the PCR to go through 20 cycles? Again, 2 to the power 20 is what you are going to use. Okay.
So in that case, it's into the 20?
2 to the power 20.
Okay, correct. 2 to the
Okay,
power.
so this is what you are going to be using. Okay. What is the difference between HBA and HBS allele of the beta globin? Okay, how does HBS call the sickle cell? There is a mutation where the nucleotide A changes. Okay, nucleotide A changes, and that's what causes it to clump up. Because of that change, what happens is the polypeptide chain folds differently, because it codes for a different amino acid. Okay, in that position, it codes for the amino acid. So here you're going to look at, it changes to valine, the amino acid glutamic acid. Oh, sorry, not here. Okay, so going back here. So it changes to valine, which is a non-polar amino acid. And because of that, the folding is completely different. Okay, so I'm going to go back to the intro part where it talks about where the mutation is and what is the results of that. Okay, so here there are many videos that kind of go through sickle cell anemia, so you can also see that. Okay, so here talking about how that changes. So this A changes to U, and because of that, in the 7th position, normal hemoglobin codes for glutamic acid. Okay, if there is a mutation, instead of glutamic acid, it codes for valine. Okay, this is another amino acid, which is a non-polar amino acid, and because of that, it misfolds and clumps up. So all of that hemoglobin aggregate and are not able to carry as much oxygen. A normal hemoglobin, which is the kind of like a donut-shaped molecule, a donut-shaped cell, and it can, each of the hemoglobin that is there, each hemoglobin molecule can bind a maximum of 4 oxygen atoms at the same time. Okay, simultaneously it can bind maximum of 4. But when it clumps up, it cannot bind as much oxygen because the non-polar part is tucked inside. And so they all aggregate because there is 90% of blood is water. And so in a polar environment, the non-polar amino acid causes it to fold up in such a way so that it does not interact with water molecule. Okay. And because of that, it supplies very little oxygen. And so the organs do not get as much oxygen, resulting in blockage and flow and oxygen deprivation to major organs. That is what causes sickle cell. And all because one nucleotide changes that codes for a non-polar amino acid. Okay, so here is a very nice description of why that happens, but I'm not going to go into that. We should continue with the questions, okay? So read up on that section so you know what the differences are and why that change causes the sickle cell disease, okay? Is everybody okay?
Yes.
Okay.
Yes.
Okay, all right. So how are we able to distinguish the HBA beta globin gene, which is the normal one, and HBS allele in our lab exercise? the mutation, because of that mutation, the mutation is at a site where it is also the recognition site for the restriction enzyme. And so if there is a mutation, the restriction site will not be recognized by the enzyme because of the change in the nucleotide. So the enzyme is not going to cut the DNA. For HBA, it will be cut by the enzyme DDE1. If there is a mutation in the HBS allele, sorry, not the enzyme, the DNA is going to be cut here for the normal, and for the mutation one, it's not going to get cut. Okay, so here, when we look at the normal one that includes, so the normal one that includes the DDE1 site, normal gene contains the DDE1 site, okay? So when this gene is, okay, so this is reacting also. So if the individual is normal, okay, and none of the mutated alleles are there, then if this gene is amplified by PCR and then digested with the restriction enzyme DDE1, then it will cut it into 2 fragments, okay? But if this one is amplified and it will produce the same fragment, 233. So if the allele is amplified by PCR, okay, we're doing PCR from these individuals, okay? They have 2 copies of the alleles, right? Each one coming from the paternal, one coming from the maternal. Because this is a homozygote, okay? The 233 base pair, all of these are going to make the 233 base pair, okay? All of these are going to make the 233 base pair. So all of these are going to be the same after you do the PCR, okay? But when you take this piece and set up the digestion with DDE1, take the PCR product and set up the reaction with digestion with DDE1, do the same thing, okay? What's going to happen is this one, you are going to get a fragment like this and a fragment like this. So you are going to get 2 fragments. Okay, so when you cut it with DDE1, if the individual is normal, you should get 2 fragments. Okay, for an individual who is a carrier, some of these will be without the recognition site. So that will be 233 base pair. Okay, but this one that has the normal allele, you will get a small piece and you will get a larger piece. So you will get 3 fragments from somebody who is a carrier. Okay, this individual, because the DDE1 site is not going to be there anymore because of the mutation, you only get 1 fragment. Okay, so depending on how many fragments you see in the restriction digestion, you can tell whether that individual is normal, carrier, or has the disease. Is a heterozygote, homozygote normal, or homozygous for the sickle cell, okay, based on fragments. So how do we know that it was cut and it has fragments? For that, we use gel electrophoresis, agarose gel electrophoresis. So agarose gel electrophoresis is used to analyze those fragments, okay? Let me show you that, and then I will come back to this.
These tubes contain identical DNA fragments, but they will be cleaved with
Okay.
different.
Can you guys hear this one or no?
Yeah.
You can?
Yeah, we actually
Yes.
Oh,
yeah, on the
I
video.
was just going to say that I should have used the headphones from the start then. Sorry. Okay. All right. That's great. I was just thinking that there are no transcribe here. Hold on. Let me increase the volume then. Okay.
Different restriction enzymes to yield fragments of different sizes. Enzyme 1 cuts the DNA into fragments A and B, which we color for tracking purposes. Enzyme 2 cleaves the DNA at a different recognition sequence, yielding fragments C and D.
So these are 3 different kinds of restriction enzymes, okay? But just to show you how the agarose gel electrophoresis works, that's what you want to focus on, okay?
Adding both enzymes yields fragments A, E, and D. Gel electrophoresis is one of the most useful means of separating and purifying DNA fragments for further analysis. In this technique, a jello-like slab of material called agarose is molded with wells, placed in a buffer solution, and hooked up to positive and negative electrodes. The DNA solutions, to which blue dye is added, are then pipetted into the wells. A well is also reserved for the placement of DNA of known sizes, and then the power supply is turned on.
The DNA of known sizes, that is called the ladder or marker. That is what allows us to find out the sizes of the unknown fragments, okay? So that's the standard sizes.
The blue tracking dye is negatively charged and migrates toward the positive electrode, as does the DNA. The DNA backbones contain negatively charged phosphate groups, which are attracted to the positive electrode. The smallest fragments move the fastest, being entangled less in the agarose matrix of the gel. When the blue dye reaches the bottom of the gel, the power is turned off. A fluorescent dye is then used to stain the DNA fragments. A DNA fragment in the size standards.
So we used a different kind of a gel where the dye was already added to the gel, and all we did is run the gel, and we were able to turn on the UV light, and the bands fluoresce. Okay, and so we were able to use these fragments, view the fragments. So these would be the DNA fragments which bind to the fluorescent dye, and when we turn on the UV light, we can see it. These are the standards which are of known sizes, and we compare how far this has gone with the distance migrated by this and can estimate the size. We can also make a standard curve using the distance migrated by these bands against their sizes. And using that standard curve, we can measure the distance traveled by this band and figure out what is the size of this band against that standard curve. And that's what you had to do for your assignment, okay? And so going back to so here is this is what the gel looked like, the one that you had, okay? And this was the marker. These were the fragments. So this was one group, 3A, B, C, and D. These were the samples from each of the 2 parents and the 2 kids, okay? And again, just based on the number of fragments, you should be able to tell who is a homozygous, who is a heterozygote, and who has the disease, okay? So remember that we talked about if the person is homozygous normal, then you should have two bands, right? The reason you would have two bands is because the normal person would have the DDE one side and it would cut, okay? If the person is homozygous for sickle cell, sorry, then the person would have one band because that individual will have, because of the mutation, will have lost the restriction site. The enzyme DDE1 is not going to recognize and is not going to cut the DNA, okay? If the person is heterozygote, meaning that the person has one of the alleles that has the mutation and the other one is normal, then the person would have three bands, okay? And you can see 1, 2, and 3 here. 1, 2, and 3 bands, right? So these are the two parents. Both the parents are heterozygote because they have the 3 bands. This individual has one band, meaning that this person must have the disease, okay? Homozygous normal, this individual has 2 bands, so that individual must be homozygous normal, okay?
I have a question.
Yes.
For heterozygote for 3 bands, does that mean that the parents, they are normal but they're carriers?
Yes, the heterozygote, the ones that have the 3 bands, heterozygote meaning they have one allele that is normal and one allele that has a mutation.
Got it. Okay.
Okay.
Thank you.
So going back to my chromosomes here. Okay. So if you look here, so this is homozygous normal. Okay. So these individuals would have 2 bands. Sorry. Would have 2 bands. Both the alleles are the same. Okay. When you amplify it by PCR, they all will have the same size fragment. And you are not going to know just by PCR. So if you just did the PCR, you're not going to know until you do the restriction digestion. Okay.
Okay.
So, okay. All right. So let's get back to the question here. So this is how we distinguish between HBA and HBS allele by amplifying the HBA gene by PCR and then cutting it with the enzyme DDE1. Okay. Okay. So here are the two sequences of the PCR primer that were used in the PCR reaction. Now, when we make the primers, we have to design the primers in such a way that it does not, it binds at that annealing temperature. The annealing temperature is when the primers bind to the single-stranded template DNA, okay? Now, these have melting temperature, the temperature at which they are going to separate, okay? And they're not going to bind to the single-stranded template DNA. Which of these, and so we calculate when we design the primers, we have to calculate the annealing temperature for each of the primers, okay? Now, typically, there are several criteria that we use to design these primers. One of the very important one is the temperature, annealing temperature, okay? And that is based on how many of these nucleotides are guanine and how many are adenine and thymine, okay? So we find the GC percentage. So all you have to do, let me see if it's gonna let me copy or not, I'm not sure. Okay, let's see. Okay, did actually. Okay, so the way you would find is count 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay, so what I did is I counted the number of G's and C's. Okay, the reason I did that is because if the primer has more G and C, then it bonds with the template DNA much stronger, because the bond between G, the complementary bond with C, is 3 bonds, as opposed to when A bonds with T, sorry, it would be 2 hydrogen bonds. Okay, so if there are more G's and C's, then the primer would anneal much better. So one of the criteria that we look at is the annealing temperature, and that's based on the GC percentage. We call it GC percentage. And if it is more than 50%, then it's a good primer. Okay, so more than 50% meaning 1, 2, 3, 4, 5, 6. So out of 20, 10 are GC. So that is equal to 50%. Okay, there is a formula to calculate the GC percentage, the annealing temperature, and that is what is given to you in your exercise, how to calculate it. Okay, and you should know that, so that if you are given a sequence of a primer, you should be able to hold on. So this is the PCR reaction, okay? If any of you find it before I do.
I think you already passes. I think it's question 3 and 4. Not
What?
sure. I think you already passed. It's question 3 and 4, I think.
Question 3 and 4?
Uh-huh.
No, we did go through that.
No, I mean, question 3 and 4 of this assignment.
Oh, question 3 and 4 of this assignment.
Yeah, that was up.
That was up?
Yeah. But I'm not sure if you're looking for that or for a formula.
I'm looking for the formula.
Oh,
to
no,
show
so,
you
right.
where the formula is.
Okay.
Okay,
No,
so there is a formula that allows you to calculate what percentage. By the way, this is something that you would have to also do. Okay, the pedigree analysis. So make sure that you know how to do this. Okay, I missed it somewhere. Hold on. Okay, I think we may have taken it out. I cannot find it, but one way to know which one would have a higher melting temperature. So what I'm looking for is which one will have a higher annealing temperature and why. That will depend on which one has more the GC percentage, okay? The first one, the one that we counted, this had 10, and this one has 1, 2, 3, 4, 5, 6, 7, and 8. Okay, so 8, and this is 10. So this one will have a higher annealing temperature than the bottom one. Okay, these are important factors to consider when we look at primers, what primers to use in the PCR reaction. Okay.
Any suggestions where to find that formula? Because I know it's been some questions of that to find the percent, but we never practice, at least in my class.
Okay, so the formula, don't worry about the formula because I cannot find where it is. It is given to you. I know it is given, but the way you can answer this question is just count. The easiest way is to just count the number of G's and C's. Okay.
Okay.
So to count the total number of nucleotides and use the number of GCs to divide it, just like I did here. Okay. So number of GC is 10, total number of nucleotide is 20. So it's 50%. Okay. If you did 8 out of 20, right? So this is a lower percentage. So this one will have a lower annealing temperature. Okay. And this one will have a higher annealing temperature. Because G and C have 3 hydrogen bonds, A and T will bond with each other by 2 hydrogen bonds. Okay, so this has a higher annealing temperature because there are more G's and C's in that primer. Okay. Does that make sense?
Yes.
Okay. All right.
Yes, Professor, that makes sense, but we have to know what G and C stands for.
Yes, you should. You should know what it stands for.
Yes.
G is for guanine, right?
And T
C is for
thymine.
cytosine, right? T is for thymine. A is for adenine. Right? These are the building blocks. There's only 4 of them. Right? Unlike amino acids, you don't have to know there's 20 of them. What is the difference between sickle cell disease and sickle cell trait? This you should be able to tell.
Well, the difference, what we know is that people with a sickle cell trait carry only 1 copy of the altered hemoglobin gene. And a difference with the sickle cell disease is that people carry 2 copies of it.
It's an autosomal recessive disorder, meaning that if it is an autosomal recessive disorder, that means both the alleles have to have the mutation. Okay, so again, going back to my drawing here, right? So if a person has a trait, then one of the alleles has the mutation. If the person has a disease, then both the alleles have to have, it's homozygous. That's the phenotype. Okay, both the alleles have to have the mutation. Okay.
Yeah.
Okay. What type of inheritance mode is this? Genotype and phenotype. What type of inheritance is this? If somebody has the trait, then the person has no symptoms, right? If the person has both the sickle cell mutations, both the alleles with the mutation, then the person has the disease, okay? So this person with the trait is not going to have the disease, okay? What are the main components of PCR reaction and what is the purpose of each? That's what I went through in the video also, and you have the link, right? What are some of the applications of PCR? It's used for identification. It's used for diagnostic, like sickle cell that we are using as an example. It's used for somebody mentioned the COVID testing, right? It's used for DNA fingerprinting. It's used for forensics. If there is a crime committed somewhere and you have a whole bunch of suspects and you're trying to find if that suspect was at the crime scene, then biological sample collected from the crime scene can be amplified by PCR and then match it with the DNA from the suspect, right? So this way we can match and see whether that suspect was at the crime scene or not. Okay, so very commonly used in forensics for diagnostics like we did, for disease, whether somebody has an infection, for many, many different things that PCR is used, okay? PCR is just a way to amplify huge amount of DNA from very little DNA, okay? It is used for identifying different unknown organisms also, okay? What are nucleases and what are they made of? Nucleases are enzymes and they are made of protein. Okay, nucleases are enzymes, they are made of protein. How does an endonuclease differ from an exonuclease? Exonuclease and exonucleases are the ones that cut DNA at the end. Okay, so if this is a DNA molecule, exonucleases are going to cut from the ends. It's going to cut from the end. Okay, it's going to, so it has to have free ends. So if you have circular DNA like this, if you have a circular DNA, exonuclease will not work because it has no free ends. Okay, endonucleases, endo means inside, so it can cut in the middle of the strand. So let's say endonuclease can cut in the middle of the strand. Endonuclease can cut in the middle of the strand. Okay, so endonucleases can cut in the middle of the strand. Exonucleases will cut only at the ends of the strand. So free ends, it has to have free ends. So if the DNA is circular in shape, then the exonucleases are not going to work. Okay, it's not going to cut the DNA because there are no free ends. But endonucleases can cut circular DNA and linear DNA. Okay. What is a restriction endonuclease? Restriction endonucleases are the ones that are restricted by where it's going to cut. It has to have a recognition sequence, and within that recognition sequence, it will cut a specific bond. Okay, it will cut a specific bond. Here in your, you have a lot of information about what restriction endonucleases are. Here, okay. So what are restriction enzymes? A special class of enzymes that cut DNA, okay? And they have to have these recognition sequence. And within the recognition sequence, it will cut a very specific bond within the recognition sequence, okay? So that's what restriction endonucleases are. Which restriction endonuclease did we use in our lab? That would be DDE1. Okay, the name of that enzyme is DDE1. You should know the name of that enzyme. Okay. Does it produce sticky ends or blunt end when it cuts the DNA? So the enzyme DDE1, this is the one DDE1, and yes, it produces sticky ends, right? Because it cuts right here, this piece and this piece. So this does produce sticky ends, okay? What is a palindromic sequence? Palindromic sequence is when the DNA, the nucleotide sequence is the same from the same end, reading from 5' to 3'. If it reads the same, then it's called a palindromic sequence. Okay, so symmetrical sequences, when it reads the same on both strands, that's what palindromic sequences are. Okay, so GAATTC, from 5' to 3' end reads GAATTC, the top strand 5' to 3' GAATTC, that is a palindromic sequence. Okay, which of the following is a palindromic sequence? So again, read from here. Okay, that would read the same.
The first one.
Yep. Actually, both these ones, this one should be a palindromic sequence. This one is actually not a palindromic sequence because the AAGGT, if you write down here, 5' to 3', AAGGTT, right? So that would be TTCCAA, right? So AACCTT, it's not the same, right?
Okay.
Okay. But if you write down the second one, GCCC, 3' to 5', it would be GGGCCC. So this reads GGCCC, right? And this one also reads the same. So this one, the second one would be palindromic, right? So this is the second one. That would be palindromic sequence. The third one, you would have to write it again, okay? So make sure before you answer, write down the other one before you select the answer, okay? The third one is AT, right? From 5' to 3', 3' to 5'. This would be TA. So when you read from this and read from this, this is the third one, also palindromic, right? The last one is not. So this one should be palindromic and this one should be palindromic also, okay? How does the number of restriction sites relate to the number of fragments produced for linear DNA? Okay, so restriction sites relate to number, okay. So if you have, let's say a circular DNA, and I'm just going to do a one line, not the double-stranded DNA, okay? So let's say that there is a restriction site here, and there is a restriction site here, okay? For enzyme EcoR1, okay? And there is a restriction site for the enzyme EcoR1, which is a restriction enzyme. If you cut this one, what you are going to get is one linear molecule, okay? So one fragment, as opposed to this linear DNA, if you cut once, you're going to get one piece like this, and let's say that this is a much longer one, then you are going to get these two pieces, okay? So you get two fragments, and here you are going to get one fragment, okay? So if the DNA is circular, if it is circular DNA, and linear DNA, both of them have one restriction site. Both of them have one restriction site. If you start with circular DNA, then the number of fragment is the same as the number of restriction sites. But if you start with linear DNA, then the number of restriction sites and the number of fragments is one more because it's a linear DNA, okay?
Excuse me, Professor, you say that for circular DNA, the number of fragments are the same of the number of, what is it?
Cutting site, yes.
Cutting size. Thank you.
Yes. Okay. So if it is linear, then you will get one more fragment plus one. So if there are 3 restriction sites, then the number of fragments will be 3 plus 1 if it is linear DNA. Okay. If it has 3 restriction sites, then you would get 3 fragments. So it's the same number of fragments. Okay. Are we good?
Yes.
All right. Okay, moving on. What is gel electrophoresis? That is something that we saw in the video. So you should be able to answer that. Gel electrophoresis is the separation of DNA, RNA, proteins in an electric field. Okay, in an electric field, the separation of molecules based on the size and charge of the molecule. Okay, the bigger size molecules travel slower, smaller molecules travel faster. So there is a separation, the size of the DNA fragments in an agarose gel, which is used for separation of DNA and RNA molecules. Okay. What does agarose gel electrophoresis allow us to do? It allows us to separate DNA and RNA fragments based on their sizes and shape. Okay. What is the chemical nature of agarose? Agarose is a polysaccharide. Okay, agarose is a polysaccharide which forms a very complex matrix, almost like a mesh of network of chains. Okay. What factors affect the migration rate of DNA through an agarose gel? The shape of the DNA molecule, the size of the DNA fragments. Okay, all of that would affect. And again,
I would say that the answer could be the size in general affects it.
The size and the shape. For example, circular DNA versus linear DNA would travel differently. Okay. And then it could also be supercoiled, much more compact DNA. A lot of DNA that bacteria has that is circular and supercoiled, that the shape becomes very compact. And even if it's the same size, it will travel much faster just because it becomes very compact. Okay. Kind of like almost twisted. So let's say that I don't know if you can actually see. Let's say that this, can you see this rubber band?
Yes.
Okay. So if it is twisted and very, very compact, becomes very packed like this becomes much more compact structure. This will travel much faster than if it is open like this. Okay. If it is cut, I'm not going to cut it, but if it gets cut and linear, then it will travel slower. So the shape of the DNA also matters. Okay.
Do you mind if you can repeat what is the chemical nature of agarose?
That is a polysaccharide. Okay, it comes from the seaweed.
Okay.
So it's actually jello-like, kind of like jello that forms a porous matrix. Okay. So this was the video that we were watching, right? Right here. So this kind of a network through which the DNA migrates. Okay. Why did we run a ladder? Okay, why did we run a ladder? Ladder is a mixture of DNA fragments which are of known sizes. Okay, that allows us to estimate the size of DNA fragments that are unknown. Okay, so for example, when we did our experiment and we wanted to find out what are the sizes of the DNA fragment, we would not be able to do it if there was no DNA ladder. Okay, so here, This is what we were looking at, right? So this DNA ladder, these DNA fragments, we can see the fragments are there, but we can also estimate the size of each of these because the ladder will allow us to do that. Okay, we know what the sizes are for each of these. And that will allow us to figure out what are the sizes of band 1, what is the size of band 2, what is the size of band 3, okay? What are some of the practical applications for the use of restriction enzyme? A lot of times these restriction enzyme, this is just enzyme that cuts DNA, okay? So when we do genetic engineering, we take genes from one organism, cut it out, and we can put it in any other organism and get that organism to make that protein or whatever that gene is coding for, okay? Very commonly used in genetic engineering to modify food, genetically modified food, for gene therapy. We can use it for diagnostics, okay? One of the hormones, have you heard of insulin?
Yes, it's very
Yeah.
famous. For people who, well, it's like when your body stops producing insulin. It's very famous.
Right, right. So insulin was actually made, is possible, and people are able to use it because of genetic engineering, because you can cut out the gene of insulin from an organism and put it in a different organism and get it to make it, okay? And so, although insulin is a human enzyme, there is a number of steps that need to be done before we can get bacteria to make it. But again, we can move it around, we can take out the gene, cut it out, and put it in a different organism and do all sorts of cloning experiments and use restriction enzyme. Restriction analysis is also used to find out, do pedigree analysis. So you want to see who is related to who and the traits that are passed on from one generation to another. We can do all sorts of things with restriction enzymes, okay? Okay, so here, which enzyme does not make sticky ends? So one of the quick ways to look at that, we have more, no, is to see where the arrow is, okay? So this is double-stranded DNA. This is the double-stranded DNA, and it shows you where the arrow is. If you see that it cuts unevenly, uneven meaning there's 1, 2, 3, 4, 5, 6, okay? 6 nucleotides, and it is cutting after the first one. It is cutting after the first one, okay? So this will be a sticky end, okay? This one, let's see if it's gonna let me do that. Actually, no. Put it all together. No, it's not gonna let me do that. But you see the arrow that this is 6 nucleotides here. So 6 nucleotides here, right? 1, 2, 3, 4, 5, and 6, okay? And it cuts here after the first one, okay? So this one, if you look at all the ones that are here, this one also cuts after the first one. This one also cuts after this one. All of these are going to make sticky end. Which one does not make sticky end? That would be this one, okay? This one, because it cuts right in the middle. So 6 base pair, and after 3, it cuts it, okay? So that will make a blunt end, which is not a sticky end, okay? All of these other ones are going to make ends like this. Okay, those are sticky ends. So the only one that's not going to produce sticky end would be this one, okay? So that's how you would figure out whether it makes sticky ends or not. Okay, so the following gel electrophoresis pattern was obtained from digestion of linear DNA molecule by two different restriction enzyme. Okay, so two different restriction enzyme. One was EcoR1 and the other one was Hindi3, two different enzymes. Okay, and so it was run on the gel. Estimate the size of the bands visually comparing the position to the DNA ladder. So this is the DNA ladder. These are the sizes. Okay, and so you can visually kind of get the idea, because if it runs, so this band is 8,000 base pair. Because this band runs the same distance as this one, this should be about 8,000. Okay, so let's take a look at, okay, let's take a look at, okay, so the way you can do that is kind of, if you take a ruler and kind of measure the distance. Okay, so let's say that you draw a reference line, and so this is about the same. So this is how much this is traveled. Okay, and this is also traveling the same distance. Okay, about the same distance. So visually, you can tell that these two bands must be about 8,000. This tells you that this is 8,000. This we don't know, but because it travels the same distance, so this must also be 8,000. Okay, not 800. Okay, there we go. Okay, so then how do you find out what is the size of this one? Again, what you can do is you can kind of see if it lines up with that band or not. You can take a ruler and kind of line it up. So it's a little farther than that. So it is a little smaller than 7,000. Okay, this one, if you line up, it's about the same. Okay, this one, if you line up, this is a little farther than 5,000. So it should be closer to 5,000 base pair. This one should be 4,000 because it's almost the same as 4,000. This band, band B, is a little farther than 3,000. So it would be smaller than 3,000, not quite 3,000, smaller than 3,000. This one, a little smaller than 2,000, okay? This one should be about the same as 1,000. So this is kind of how you would figure out what are the sizes of each of these bands, okay? So that's how you would find out the sizes of each of the band. Let's look at the question, what else it is asking. So that's visually comparing the position to the ladder. That's how you would do it. Just place a ruler and see how far it goes. Measure the distance of the migration of all the ladder bands plotted against their size on a semi-log paper to make a standard curve, okay? So we do a standard curve. This is something that you had to do for your assignment also, where you're given the sizes of DNA fragments, and you would have to measure the distance and come up with a standard curve, okay? And match the distance of migration to get the sizes. How many times does EcoR1 cut and Hindi3 cut this DNA? Okay, so lane number 1 has DNA. It's all of it is the same DNA that is cut with different enzymes. Okay, so lane 1 has DNA that is cut with Hindi3. So lane 1 has DNA that is cut plus EcoR1. Okay, so this is cut twice. You are getting 2 fragments. Okay, so whatever the DNA is, EcoR1 is cutting it. So this is about 5,000. Let me wait. Okay. So this is about 5,000 base pair because it is closer to that. And this is about 3,000 base pair, okay? So whatever this size is, 8,000, you add up these two fragments and somewhere here, the echo R1 is cutting, okay? And so this piece is 3,000. And this piece would be 5,000, okay? So this DNA, when it is cut with echo R1 over here, it's producing two fragments that are of this size. Hindi 3 has a different recognition sequence, and it is going to cut at a different site, and one piece is 7,000, and the other piece is so 1,000, not 1,000. So 1,000 plus 7,000 is still 8,000. So either it is cutting here where one piece is 1,000 and the rest of it is 7,000. So the rest of it is 7,000, or it could also be here, but you can't tell based on this, okay? So this is called restriction mapping. We can map the position of these enzymes. So this would be Hindi 3. And there could be multiple correct ones, okay? There could be multiple correct ones. So we can find this out by looking at, this is called a double digest. Okay, double digest meaning two enzymes, EcoR1 and Hindi 3, both of these are added to the same tube. So tube number 1, tube number 2, tube number 3, number 4. So tube number 1 had DNA plus EcoR1. Tube number 2 had DNA, same DNA plus Hindi 3, and we do the digestion. Tube number 3 had DNA plus EcoR1 and Hindi 3 both in the same tube. Okay, that's called a double digestion. It's both the enzymes are added to the same tube. Okay, and this one is uncut DNA, meaning that the DNA was not cut. No enzymes were added, no restriction enzyme. So this is an uncut DNA. Okay, so this is 4,000. This is 2,000. That does not add up. This is 1,000. This is about 2,000. So that's about 7,000. That does not add up. It should be 8,000 total. Okay. And that allows us to figure out what the site is. Okay. So this is kind of what it is. The figure, this should be a little higher. This should be 5,000 and not 4,000. Okay. So this is what this question is asking you. How many times does EcoR1 and Hindi 3 cut this DNA? Hindi 3 cuts it once and EcoR1 cuts it once. Okay. How do we know that? Because when you cut it, the linear piece of DNA and you are getting two fragments, 1 and 2 fragment, when you cut it with Hindi 3, you're also getting 2 fragments. Okay, different size fragments, but you are getting 2 fragments. Okay, which means that when a linear DNA is cut once, you get 2 fragments. Whether it's cutting here or here, wherever it's cutting, you're getting 1 and 2, 1 and 2 fragments. Okay, so when you're getting 2 fragments here and 2 fragments here, that means both EcoR1 and Hindi 3 are cutting the DNA once. Okay, at different sites, but only once. Okay.
Wait, so you said both of them produce 2 fragments, right?
Right. So if you look at lane 1, you are seeing 1 and 2 fragment. Lane 2, 1 and 2 fragments, right?
Yes.
So because both of them are producing 2 fragments, that means both of them cut once.
Mhm.
Okay. Okay. Okay, so you may not have to do the standard curve because you have to go to the stations, but you should know how to use the standard curve. Okay, you should know how to use the standard curve. And let me see if I can pull up a standard curve. Okay, now it let me do it. Okay, so here you have a standard curve where this is the distance traveled by these vans. Okay, let me move it up. So these distances that are traveled by each of the band, and it is plotted against the size of the DNA. Okay, so when we are looking at unknown fragments, so if we wanted to measure the size of this, we would measure the distance traveled by each of these bands, put it on the graph, and then see where it intersects and figure out the size from there. Okay, so we would use the ruler. So you will be given a ruler, so you can do that. So you would do it the same way. Oh, no. So you would use the ruler, measure the distance, whatever, and you measure it in millimeter. Okay, so if you look at the units, it's in millimeter. So you would measure using the millimeter side of the ruler, measure how much this is, and then find that point on this y-axis, draw a line. So what you would do is place the scale here. So whatever, let's say this is how far it traveled, and you would draw a line here. Okay, and then see where it is intersecting that one, okay? And then see where it intersecting this line here, and that's how you would read it, okay? So this would be a little over 4,000. So whatever distance that it traveled, this is how you are going to use the standard curve, okay? This is how you're gonna still use the standard curve to find out where, how big the size is. So draw a line from here and read it off of the x-axis. So whatever that size is, that's what is going to be the size of the band that traveled this much, okay? So you are not going to have to draw the graph, but you will need to use it, okay? Okay, so another 2 families were screened for sickle cell disease using the same PCR primers and DDE1 digest as we did in our lab. Determine the genotype and phenotype. So I think that you should be able to do this one, right? We have gone through enough of the sickle cell genotype and phenotype, heterozygous carrier, homozygous normal, homozygous affected of the parents A, B, C, and D, right? It's just like the one that you did in your lab.
Yeah, it's true. Something similar.
Right?
Yeah.
What do you guys think? You guys okay?
Hello.
Yeah, we okay.
Yeah.
Getting longer, but it's okay.
Okay, okay, because it's already 10. That's why I'm.
Yes.
Okay.
It's already 10.
Yeah.
It went by fast though, to be honest. But it felt that it went by fast.
Okay. So I think that this is the last part of PCR, and I think you should be able to do this 2 different families, and you would have to determine. Oh my God. Okay. You would have to determine whether the individuals have sickle cell anemia or not, okay? Or who is a heterozygote, who is all of that, right? The parents are A and B, the children are C and D, and it's the exact same kind of example that you have, okay? So again, you can use the same analysis that we did, 3 bands, 2 bands, 1 band, and how you can tell which one is normal, which one is a heterozygote, right? So here, 3 bands meaning what? Right? So if it's homozygous normal, then have 2 bands. If it is sickle cell, then the individual has 1 band. If there are 3 bands, that means it's heterozygote, right? So you can use the same analysis for these people, right? 3 bands, 2 bands, based on that. So I think you should be able to do it. Okay?
Yeah.
Okay.
All right then. Let's look at the transformation experiment, okay? For the transformation experiment, what you had done is you were given bacteria, and the bacteria was made competent. Competent, what that means is basically what you did is you added the DNA and got the bacteria to pick up that DNA in the cytoplasm and express the genes that were on that DNA, okay? The DNA that is used is a plasmid DNA, okay? It's a plasmid DNA, meaning that these are small extra chromosomal DNA, okay? So some bacteria, let's see, I have it. Okay, I'm gonna have to move this. So bacteria have this chromosomal DNA. That's called the chromosomal DNA. This is the chromosomal DNA or the genomic DNA. Okay. So this is a bacteria that has this genomic DNA. And they can also have DNA that is not part of the genomic DNA. These are extra chromosomal DNA. These are very small circular extra chromosomal DNA. Okay. And these are called plasmid DNA. They also carry genes, but very little. Okay. So these are called plasmid DNA. What you did in your experiment, you were given bacteria that did not have plasmid. Okay. And then you added plasmid DNA. So this had a pure solution of plasmid DNA. You added to that. Okay. And then we did what is called transformation. Transformation is uptake of DNA from surrounding environment. Okay. Meaning that in the tube, if there is DNA and the cells take up that DNA into the cell, and so now the cells, some of the cells, not all of them, will pick up that DNA, that small DNA, okay, that small DNA, they will pick that up and they have the chromosomal DNA. Sorry. So they have their chromosomal DNA, okay? And some of these cells have taken up the DNA, okay? And so this cell took up the DNA, this cell did not. Okay, so in the same solution, you can have a fraction of the cells that picked up the DNA, some of them did not pick up the DNA. This process of picking up the DNA, that is called transformation. Okay, that is called transformation. And I will show you. The experiment that you did here. This is what you had done. You had 3 different tubes, okay? You had 3 different tubes and you added, this is the name of the plasmid DNA. This is the tube where you did not, this was a control that you did not add any DNA. And this was another control where you added just water, okay? Now notice the word competent here, okay? Competent means that these were cells that were treated with chemicals so that they have these tiny pores in the membrane. So these are not tiny, but there are these very small pores in the membrane that are made by treating the cells with calcium chloride, okay? Calcium chloride is a chemical that makes these tiny little pores in the membrane so that when DNA is added, it can go in to the cell, they can enter the cell, okay? Along with 42 degree, which is called a heat shock step. Heat shock, that by changing, so this is 4 degrees, we change it to 42 degree for a very short time, and then we put it back on ice. So changing the temperature kind of shocks the cell, which speeds up the process of these cells taking, this is the name of the bacteria, E. coli is the name of the bacteria that will take up the DNA, okay? Let's see. This is gonna...
Partner with us. It's not just the journey.
Edvotech instructional videos. Transformation. For this experiment, you will need an E. coli source plate.
Okay, hold on, not this one. Let me.
The goal of transformation is to
Okay. So this is the one that I want you to look at.
cause bacteria, in this case E. coli, to pick up DNA plasmids from their environment. The bacteria are negatively charged because of the structure of their lipid bilayer cell membrane. And the plasmids are negatively charged as well because of their DNA's phosphate backbone. Since they are both negatively charged, they tend to repel one another. To facilitate this interaction, calcium chloride is mixed in with the bacterial cells. Once in solution, the positively charged calcium ions bind to the negatively charged phosphate groups on the cell surface and neutralize their charge. Plasmids containing the gene of interest, RFP, are then mixed with the treated cells at 4 degrees Celsius. The temperature is quickly increased to 42 degrees Celsius, and then reduced back to 4 degrees Celsius. This heat shock results in the formation of adhesion zones, creating a leaky plasma membrane. This may enable the cell to take up a plasmid from the solution. After heat shock, the bacterial cells are placed at 37 degrees Celsius and allowed to recover before being plated. Bacterial samples are now put on agar plates to test for successful transformations. The plate on the left contains the antibiotic ampicillin alone, and the plate on the right contains ampicillin and the sugar arabinose. On the left is a negative control plate, P-, where the transformation reaction was done without any plasmid. In the middle is another negative control, where transformation was carried out with plasmids, but where the gene of interest will not be expressed since the growth medium lacks the arabinose inducer. On the right is the permissive growth condition with arabinose that will let us identify successful transformations. After a 24-hour incubation period.
So ours was slightly modified from what you are seeing here. And so what we did is when we incubated these, we took samples from here and we incubated them on plates. Okay, so 3 different plates. So one of them was plate A, okay. Plate B, which we expected no growth, and then plate C, which has a lawn of bacteria. All of this bacteria is growing on this. Okay, so 3 different plates. So these ones are individual colonies. These are called colonies of bacteria. This one has no growth, and this one has a lot of growth. This is what we expected. This is coming from tube A, which had the P-glow plasmid added. So some of the bacteria that picked up the DNA is growing on it. Plate B, which was on a plate that contained an antibiotic that kills the bacteria, okay, if the bacteria is sensitive to it. So one of the terms that you should know what that means is ampicillin. Anybody heard of ampicillin?
Yeah.
Yeah. Ampicillin is an antibiotic, okay, that kills bacteria. Okay, it kills bacteria, but not all bacteria. Okay, it will kill bacteria if it is ampicillin susceptible. Ampicillin susceptible meaning that it can be killed. Okay, but some bacteria are ampicillin resistant, meaning that they will not be killed because they are resistant. Okay, so how are they resistant? These ampicillin resistant bacteria make an enzyme, beta lactamase. Okay, name of the enzyme is beta lactamase, have the gene, have the BLA gene. that codes for this enzyme, okay? And so they will break down the drug ampicillin before the drug kills the enzyme, kills the bacteria, okay? And so they have to have that gene in order to be resistant. This gene, the blood gene, is present on that P globe plasmid, has blood gene, okay? So if the bacteria picks up that gene, it also becomes resistant to ampicillin. That's why this plate that has ampicillin, the bacteria that became transformed are growing on this plate. But this one, we only added water, so it does not have the gene, and so nothing grew because the plate has ampicillin added to it, okay? This one does not have ampicillin, okay? Plate C does not have ampicillin. So this is just LB. LB is the general food. This one is LB plus ampicillin. This one, these plates are just food for bacteria, okay? Plus ampicillin also, okay? So this one will allow growth of bacteria only if they have acquired the blood gene. Otherwise, they are not going to grow, okay? Now this plasmid also has a gene for green fluorescent protein, okay? GFP, or green fluorescent protein. Let me get this away from here. So this one, uh-oh, I'm getting, let's go here. My computer is freezing. Okay, so here, the 3 different plates, okay? A, B, and C. These 3 plates, this one has ampicillin, okay? Plate A has ampicillin. So this is plate A. This is plate B. And this is plate C. This one also has ampicillin. This one does not have ampicillin, okay? Ribinose is a sugar that is needed for the bacteria to make the GFP protein, okay? So that is present in this one, but this doesn't have it, and this doesn't have it, okay? The P globe plasmid was only given to tube A. This one didn't have it, and this one didn't have it. Water, this one we did not add water. This one we did add water, and we added water to this one, okay? So this is what the plates have, plate contents, okay? This is what the plates had. Now growth, expected growth, we expected colonies of bacteria on this one. We did not expect any growth. This is a control, and this was a lawn of bacteria that was expected, okay? That everything will grow because it did not have any antibiotic in this plate, okay? This is the antibiotic that will kill the bacteria unless the bacteria has the gene that breaks down the drug ampicillin, okay? And so here you are going to find that explanation that it's going to make that green fluorescent protein, okay? And the resistance gene that makes it resistant, okay? That the plasmids are these small self-replicating circular DNA, okay? And the antibiotic resistance gene, this is the ampicillin R gene that is present on the DNA, and it is going to need these green fluorescent protein in order to glow green, okay? And so if you expose this to UV light, this is how the colonies will fluoresce, okay? So this one right here, which also had the colonies growing, but it did not fluoresce here because we didn't expose it to UV light. But if you expose the same plate, it's the same plate. If you expose it to UV light, the colonies fluoresce green because it makes the GFP protein, which is the green fluorescent protein. Okay, so that's the green fluorescent protein. And for the bacteria transformed with P glow and the media with arabinose sugar and ampicillin, it will express the GFP protein and will fluoresce green. Okay, and so you can see the video here. This is a very nice one. Okay, this is a nice mix and match chart that will help you go through all of that. So this LB agar, oh, it's kind of freezing up on me. LB agar is nutrient used to grow bacteria, such as E. coli. Okay, so that would be 2. Okay, there is 2. Okay. Glycerol is something that is used for freezing the cells. Okay, so that is going to use as a cryopreservative, preserves, prevents formation of ice crystals and protects. So that would be 5. So let's see if it's gonna let me, okay, there. Okay, so ampicillin is an antibiotic that kills bacteria. So that would be 6, okay? My computer is kind of slowing down, and so I'm just going to tell you the numbers here. So C would be 6, okay? This would be 6. I'll see how far I can get it to work. See how long it's taking for the 6 to appear. Ampicillin R gene is the gene that codes for resistance to codes for the enzyme beta lactamase. Okay, so that would be number 12. D would be 12. Okay, so there. Okay, so D would be 12. Calcium chloride will make the cells competent, which would be number 3. Okay, so that would be number 3. Beta lactamase is an enzyme that breaks down the drug ampicillin. Okay, so that would be number 7. Okay. Plasmids, these are small circular extrachromosomal DNA that is self-replicating. So that would be 1. P-glow is a plasmid that has the beta lactamase gene. Okay, so P-glow is a plasmid, will make beta lactamase and grow on LB agar. So that would be 10. Competence is the ability to take up DNA from the surrounding environment. So that would be 4. Transformation is uptake of DNA. Here, 8. Okay, ampicillin resistant E. coli means that it cannot grow. Ampicillin sensitive is the one that cannot grow, so that would be 11. Ampicillin resistant would be the one that breaks down the ampicillin. Okay, so it should be antibiotic that kills, that is ampicillin. Yeah. Ampicillin resistant E. coli means that it can grow, will make beta lactamase, and it can grow in the presence of ampicillin. Okay. So that would be 10. Okay.
We have 10 twice.
Sorry?
We have 10 twice. And we don't have 9.
You don't have what?
We
For
have 10
H?
twice.
Yes, we have 10 twice. And we
Oh,
don't have
10
9.
twice.
Yes.
Okay, where do I
For
have
H,
10?
for H, for
Oh, for
P.
HP
It was
glow.
10.
Okay, let's see. P glow.
We don't have 9. So
Yes, we don't have 9.
Which one do we don't have?
9.
9. Okay. So 9, yeah, this could be 9. This could be 9. Plasmid that has BLA gene, ampicillin resistance gene. Okay. So that would be, and what did I say for 10? We'll have beta lactamase and will grow on. So this would be 10. That's fine. This would be 10, but this could be 9. Okay.
Okay.
Okay. Yes. Thank
So
you. Good
what,
catch.
what is going to be 9? Sorry.
So H would be 9.
Okay.
Okay. H would be 9. That is plasmid that has BLA gene. Okay. And it could be actually both P glow, because P glow is what is going to make it resistant to ampicillin. Okay. So both of those would be correct for these. So 9 or 10 would be true for both of these, because E. coli is resistant because of the plasmid P glow. Okay.
Okay.
So sorry, let me do it here so you can kind of. So the plasmid P glow has genes for resistance to ampicillin. Okay, which means that the bacteria that gets the plasmid will become AMP-R. Okay, that's 1. 2 has genes for GFP, which is the green fluorescent protein. Okay, so the bacteria will fluoresce green. Okay, so it has, if P glow has both of these genes. So if the bacteria gets the P glow gene, it will be resistant, and it will also fluoresce green if it is grown on a plate that has the arabinose sugar also. Okay, so if the bacteria is grown on a plate that has ampicillin and arabinose, so plate has ampicillin plus arabinose. Okay, that's when it is going to fluoresce green. If it does not have arabinose, then it will still glow, it will still grow, but not fluoresce. Okay, so if it only has ampicillin but no arabinose, then it will grow on it, but it will not make the GFP. Okay. That's what, here. That's what this was talking about, okay? So it will fluoresce and express the GFP protein, and it will fluoresce, but only if the plate has both, okay?
Okay. Yeah.
Okay, so what is the link between P-glow and ampicillin resistance? P-glow is the plasmid that has the genes for ampicillin resistance, okay? How did we know that our transformation experiment worked? We look at the results from the experiment, and if you have colonies growing on plate A, nothing growing on plate B, and a lawn of bacteria on plate C, okay? So here I'm going to go back to this result, okay? So if there are colonies here, no growth here, and lawn of bacteria. Lawn of bacteria here, then that means the experiment worked, okay? Fluoresce green, yes, here it will fluoresce. No growth, so there is nothing, okay? And here will be no, okay? So if this matches with what we got, then the experiment worked, which means plate A, if there are colonies like this, if there are colonies like this, so these ones that have colonies, see these ones, these have colonies. Plate A has colonies, plate B has nothing, no growth, and C has all of these fuzzy growth, okay? So that means that the experiment worked, okay? So how do we know that the experiment worked? If you have a lot of growth like this, colonies like this on plate A, and nothing, no growth here, okay? No growth here on plate B, a lot of growth, the whole plate is covered with bacteria, and there are these colonies, that means your experiment worked, okay? So fill the table with what you observed, that depends on your experiment. Transformation occurred on which plate? That would be plate A. What evidence do you have? Because it has grown in the presence of ampicillin and is glowing, fluorescing green, okay? Which plates have glowing growth? That would be plate A. Explain the causes for bacteria to glow. Because it has the plasmid has the genes for GFP, that's why it is making the protein, green fluorescent protein, and is fluorescing green, okay? What is the role of plates A, B, and C? Plate A is the experimental
Professor,
plate.
I think this is the question for the lab assignment. This.
Oh, thank you. Actually, we have similar questions here. How do we know that the experiment worked? Because the plate A has colonies of bacteria growing, plate B has no growth, and plate C has a lawn of bacteria, okay? The controls were plate B and C. Okay, what were the control? Plate B and C. Plate B to make sure that the bacteria was killed by ampicillin, so that when we transform and it has acquired the gene, only then it will be able to grow in the presence of ampicillin. Okay, so that's why we had plate B with ampicillin, to make sure that it was not already resistant. Okay, plate C was to make sure that the bacteria was alive. Okay, the bacteria is alive, and that's why we did plate C. Okay, so the two plates that are controlled, this is a control and this is a control. This was to make sure that the bacteria is susceptible and is killed by ampicillin, and this one to make sure that the bacteria is alive. Okay, so that this is the experimental plate, and we know that the experiment worked. Okay. Why did we use these, and what does each control tell us? That's what the controls tell us. Okay, that the bacteria 1, B will tell us that the bacteria is susceptible to ampicillin, and C will tell us that the bacteria is alive. Okay. Okay, so why do bacteria survive if they accept the pea glow from outside? The bacteria survives because the pea glow plasmid also has ampicillin resistance gene. Okay, has the ampicillin resistance gene, and so that's why they survive. So these plates that have ampicillin arabinose and plated with this one.
So what was the reason why they survived again, Professor?
They survive because ampicillin resistant. Hold on. Colonies of ampicillin resistant E. coli. So that's what it would be. They survive because the pea glow plasmid has the resistance gene for ampicillin.
Because the plasmid has
the plasmid has the gene for ampicillin resistance.
Ampicillin
Okay.
resistance.
So this one should be no growth. Okay, so this is plate B, should be no growth. This one should be okay. So this one should be no growth. Again, my computer is really slowing down. So this should be lawn of bacteria. Okay, so if you find it, so this is what it would be. What?
Yeah, what is called bacterial?
Lawn.
Lawn, okay.
Okay. So that's what it is called. It's not showing up. Oh, there, finally. Okay. All right.
All right.
Okay, then. Okay, this one should not be very difficult. You have done a lot, but what I want to focus on this one, we are already 10:35, and I'm getting these notifications that in 24 minutes, my computer is getting updates, okay? So I will not have a choice but to stop. So what I want to do here is I'm going to go to this one, okay? Because what you are going to have, you should be comfortable with what mitosis is and the different stages of cell division, right? The 4 stages of cell cycle, right? G1S, G2, and M phase, right?
Shouldn't it be the PMAT?
What?
Shouldn't it be prophase, metaphase, anaphase, and telophase?
Cell cycle is the life cycle of cell.
Oh.
Okay, cell cycle is the life cycle of a cell. The stages of mitosis are the prophase, metaphase, anaphase, and all of that. So different phases of mitosis. Okay?
Okay.
So mitosis and cell cycle are not the same. So make sure that you understand. If you're not sure or just want to clarify, ask the professor.
Okay, I will ask him.
So list the 4 stages of
cell cycles be like G1.
Right. So if you look at the cell cycle is G1, G2, S, M. This is the cell cycle. Okay? This is the cell cycle. The phases of mitosis, that's where the prophase, metaphase, anaphase, and telophase are. Okay?
Okay.
So mitosis and cytokinesis, again, when you go to the stations, you are going to be looking at images and have to identify what stage of mitosis is it at? Is it a dividing cell? Is it a non-dividing cell? Okay, so those would be the kind of questions that you are going to get. And so when you look at the images, you should be able to tell what stage is it at. That's why I want to go into this one. Okay, so let's see if I can magnify it. Okay, so this is different stage, different cells looking at different. So when you are looking at it, see there is a cell plate forming. What is that? Again, my computer is freezing up, and so I'm not being able to read all of it. Can you guys see it? I am not being able to, and it's not gonna expand it for me also. So cytokinesis and telophase, if there is an option for cytokinesis and telophase, that's what it would be. Okay.
It's telophase.
This would be telophase, and this is what? Is it telophase / cytokinesis?
Yes.
Okay, so that would be this one. Okay. Why? Because you can see the telophase and also cytokinesis.
Mm-hmm.
Okay, so it would be both simultaneously.
Yes.
So typically at the last stage of mitosis, cytokinesis begins. These are plant cells, and plant cells have the cell plate forming at metaphase plate. And so that one would be a plant cell. You should be able to look at that and be able to tell from that. Okay. Can you tell this one?
Not sure, it's small, but look like it's metaphase.
This would be pro-metaphase. This is not metaphase. This would be pro-metaphase. Metaphase is actually very easy to tell because it would be all lined up right at the center like this. Okay, that would be metaphase. That would be metaphase. Okay, so this one would be pro-metaphase. What about this one? This one would be pro-metaphase also. Okay, pro-metaphase is when it's not rounded like this. This is prophase. This would be prophase. Okay. This one is also telophase / cytokinesis. This one would be metaphase. Okay. This one would also be telophase. This is what?
Anaphase.
Yes, this would be anaphase. This one is also telophase. This is also prometaphase. Okay, so this is something that you want to practice and you want to make sure that you have the right one. Okay.
Mm-hmm.
Okay.
And again, you have lots of figures to practice from. Okay, this would be anaphase. Right. So practice with these ones, look at your text and try and solve these ones. let's look at a few more minutes. List several attributes of mitotic cell division that provides to multicellular life. Growth and development, reproduction, tissue repair, all of those are possible because of mitotic cell division, because it creates more cells, right? So you should know all of this from chapter 12. No, yes, chapter 12 is where we talked about mitosis, right?
Yeah, there's where we talk about mitosis and
Right.
yeah, mitosis and yeah, I was gonna say meiosis, but that is not included. Yeah.
No. Meiosis is for making reproductive cells, gametes.
Yeah,
Okay?
again.
Yep. Okay, so this is another question that we probably didn't talk about in chapter 12. Why are onion root tip and whitefish blastula useful tissues for the study of cell division?
I think it's because you can see the different, you can see in the same, in the root tip, the different cells division in mitosis.
Right. So because onion root tip is very actively dividing and growing and getting longer and longer in size because it's getting deeper into the soil. So a lot of cells are going through division. So when you want to study the different stages of cell division, you want to use a tissue that is actively dividing, okay? And onion root tip is a good example for that, good model to study for plant cells. Blastula is embryo for whitefish, okay? And so that's also very actively undergoing division. So if we want to look at different stage of animal cells going through mitosis, embryo cells are useful tissues for that, okay? That's why we use these tissues. For animal cells, we use whitefish blastula. For plant cells, we use onion root tip, okay? What are the genetic consequences of mitotic cell division? Genetically identical as the parent cell. Okay, so the daughter cells are genetically identical to the parent cell. Know the number of chromosomes of the physical state of chromosome in each stage, right? So these are something that you should know, right? Know the number of chromosome and the physical state of chromosome in each stage. Prophase, metaphase, anaphase, telophase. What happens there? The chromosomes are together until anaphase, the duplicated chromosomes separate, right?
Yes.
Okay, so the number of chromosomes are counted by the number of centromere. As long as they are attached, they are counted as one chromosome, okay? So something to keep in mind. Looking at, we went through mitosis and cytokinesis. Mitosis is the division of the nucleus.
Professor,
Division.
do you mind for question for B?
For B, this one?
Yeah, no, next one, down.
This one? Okay.
Yeah, in human cells.
Okay, so human cell has 46 chromosomes at G1 phase. How many chromosomes will it have at G2?
So they multiply.
They will have twice as many chromosomes, but the chromosomes are going to be attached, okay? So at prophase, it will still be 46, metaphase, it will still be 46, anaphase, it will be twice as much, okay? And then telophase, it is going to separate because what happens in here, oh, actually that, we do have images, hold on. Okay, so when it is at prophase, the chromosomes, see the chromosomes are still together. So we count it, it will be duplicated, but they're attached. Okay, so it will still be 46. Okay, when it is at prophase. When it is at metaphase, it's still attached. So it will still be 46. But in anaphase, the sister chromatids had separated. So now it will be one cell with twice as many chromosomes, but it has separated. Okay. In telophase, it is separated. The two nuclei, daughter nuclei are forming, and it will still have twice as many. So 46 times 2 chromosomes, but the cells are eventually splitting up. And so the daughter cells are going to end up with 46 chromosomes, each of them. Okay. So in anaphase and telophase, there will be twice as much, but up until metaphase, it will still be 46. Okay. So in G1 phase, it will have 46, G2, it will be 46, but in the M phase, prophase, metaphase, it will also be 46. Okay. Anaphase and telophase is when it will have twice as much, but then after cytokinesis, it will go back to being 46. Okay.
Okay.
Okay. So what is the difference between mitosis and cytokinesis? Mitosis is nuclear division. Cytokinesis is cytoplasmic division. Okay. How do plant and animal cells differ in the execution of cytokinesis, and why?
Plants has the wall.
Very good. Plant cells have a cell wall. Animal cells do not have a cell wall. And so when they carry out cytokinesis, it forms a cleavage furrow. The plasma membrane kind of pinches in and separates the two daughter cells. Okay. In case of plant cells, it does not pinch in and separate. Instead, a cell wall forms with the vesicles and Golgi apparatus and all of those line up and start to build the cellulose, which makes up the cell wall, and eventually they separate. Okay. So because of that, the only part that is different during cell division is the cytokinesis for plant cells and animal cells. We looked at two different tissues, right? The plant cells, these are the plant cells, and these are the animal cells. All of the stages of mitosis are the same. The nuclear division is the same, but cytokinesis is what is different between the two tissues. Okay.
Yeah.
Yeah. All right. There was another one that I wanted to share with you. Let me give you the link for that. If I can find it. Now I have lost it. In all of this, we went through a lot.
I know, this is a lot.
Yeah, it is a lot.
Oh my gosh, yeah.
So are we almost done?
I think so. We are done.
Perfect.
Right?
Wow.
Yes, it's a lot.
Yes. But good luck to you guys, and I hope it was worth staying the entire time.
Yeah, it was worth it though. Though there were many exercises that I do not have in my lab exam to review, but I learned a lot.
Okay. Okay. Thank you. And I hope that you all get an A.
We hope to.
We hope.
But to be honest, it's a lot to cover in the lecture, and also I feel it's like two different courses related, but a lot of details to cover. So it's hard to keep up everything.
Yeah, especially for people who do not speak English as their first language, it's a lot of work.
Well, that is true for a lot of people, including myself.
Yes, definitely.
Yeah. So yeah. So that is definitely true. And I also agree that the lecture and the lab component are very different, but it just is the nature of this course that there's nothing we can do, because this is one of the courses, the foundational course that gets transferred to College Park, and their course content is very rigorous. If we change the rigor of the course, you may have to pay twice as much, 3 times as much to take the course over there instead of having it transferred from Montgomery College to College Park. So that's the reason why we can't do anything about it.
Yeah.
Okay.
Yeah.
So, okay.
Thank
well,
you. Thank you so much for your time and your patience. Appreciate it.
Thank you.
You are very welcome. Very welcome. Okay?
Yes, thank you so much.
You're welcome. Have a good night. Relax, and then tomorrow go over everything. I will, so hopefully by tomorrow, I will get the recorded session, and I will share it with you guys, okay?
Okay, thank you. So our professor should share it with us?
Yes, Yes, so I will be sending it to the course coordinator, Dr.
Yes.
Castanos, and
Uh-huh.
she will give it to all of the course, all the instructors. Okay?
Okay, wonderful. Thank you so much.
You are very welcome.
Thank
Okay?
you.
Bye.
Good night.
Good night. Good night.
Good night, Professor.
Bye.